Uh, Good morning, everyone. On the behalf of MIT Institute of Organic Agriculture, MIT University, Noida, I, Dr. Nikhil, Assistant Professor, extend a very warm welcome to all the distinguished experts, scientists, faculty members, researchers, and participants joining us for day two and session one of the 5-day faculty development program on climate resilient agriculture and allied technologies for net zero and socially inclusive framework. I would like to express our sincere gratitude to our visionary leadership for their unwavering support and encouragement. We are deeply thankful to Honorable Founder President Dr. Ashok K. Chauhan Sir, Honorable Chancellor Dr. Atul Chauhan Sir, Honorable Vice Chancellor Dr. Balvinder Shukla Ma'am, Director General MIT Food and Agricultural Foundation Dr. Nutan Kaushik Ma'am, and Director MIT Institute of Organic Agriculture Dr. Dioli Sanyal Ma'am, whose guidance and commitment have made this program possible. As we begin day two of faculty development program, it gives me immense pleasure to continue this academic journey dedicated to capacity building, knowledge exchange, innovation, and excellence in sustainable and climate resilient agriculture. Today's session bring together eminent experts who have made remarkable contribution in the fields of plant pathology, agricultural biotechnology, and agricultural engineering. Their insights will undoubtedly enrich our understanding of emerging biotechnologies and strategies for achieving a net zero emissions and socially inclusive agriculture future. I once again welcome all the participants and resource persons and wish everyone a productive and enriching learning experience. So, first I would like to introduce our today's first speaker, Dr. Harikesh Bahadur Singh sir. It is my privilege to introduce our Dr. Harikesh Bahadur sir, an internationally renowned agricultural scientist, plant pathologist, and NAS fellow. Dr. Singh has over 4 decades of experience in agricultural research and innovations, particularly in biopesticides, biofertilizers, microbial technologies, nanotechnology, and sustainable organic farming. He has served several prestigious institutions, including Banaras Hindu University, NBRI, CMAP, and GV Pant University. Currently, he serves as Distinguished Professor and Director of Somvanshi Research Foundation. He is the recipient of numerous prestigious honors, including MS Swaminathan Award, CSIR Technology Prize, Vigyan Ratna Award, and several Lifetime Achievement Awards. We are honored to have him with us today. Sir, I kindly request you to deliver your lecture. Please, sir.
Thank you, Nikhil, for a very nice introduction. Yesterday, in fact, I was listening all the lectures, very curious mind. And I was very happy that most of the speakers, they were talking on modern science, particularly the genomics and metagenomics, proteomics, all kind of omic technologies. My lecture will be totally without any omics. But you will appreciate that whatever we have contributed in the last 25 years from different places, they are directly related to the farmers and farming community. And to be honest with you, this will be very, very useful to all of you, particularly those who are working on organic agriculture. So the topic of my lecture is management of soil-borne diseases using microbial pesticides. Are you able to see the screen? The screen is visible?
Yes, sir. It is
Okay.
highly much visible.
Okay, thank you, dear. So now the question is that what are microbial pesticides? For the beginners, particularly the students, they must know that any biological agent such as bacteria, fungi, viruses are their components which can be used to kill or suppress the growth of microbes is known as microbial pesticide. There are three distinct types of microbial pesticide. One where the direct live microbes are used. The other one where you are using the secondary metabolites of the microbe. And the third one, of course, they are botanicals. I'm not touching the botanical part, but of course we have done a lot of work on botanical also. But here in this particular lecture, I will be focusing only on microbial pesticides. Now, friends, you should know that what are the different kind of microbes which are used globally and particularly in India. And before that, we must know that how much losses are caused by different pests in India. If you see, according to a estimate by ISOCAN, that about 2 lakh crore loss is there every year because of the different kind of diseases and pests. And if you see the number percentage wise, diseases, they are approximately causing 25% loss, insect 23%, storage 10%, rats 8%, and weeds 28%, and other 6%. So altogether, 2 lakh crore losses are there because of the different kind of pest attack on crops. Now these crop pests, they are controlled by different chemicals. As you know that pesticides are normally used for controlling different kind of pests. And in pesticide, fungicide, herbicide, insecticides, they are the one. And biopesticide, on which I will be focusing, they are contributing only 4% of the total plant protection chemical used in agriculture in India. So extremely sorry to tell you that many scientists all across the country, they are working on biological control for the last 40 years. Large number of products are available in the market, but still the percentage of biopesticide used by the farmers is very, very low. And therefore, I think meeting like this, we have to encourage the farming community that you should use biopesticide for managing different kind of pests. Now, friends, see the situation today. The biopesticide business is only 9.9 billion US dollars. This is a global database, I'm just telling you. And as compared to the chemical pesticide, which is 147.71 billion US dollars, approximately 150 times more use of chemical pesticides for managing different kind of pest disease in our country. We all know that biopesticides are eco-friendly, and chemical pesticides, they pollute soil, water, and air. But still, majority of the people, majority of the farmers, not only in India, all across the world, they are using chemical pesticide for managing different kinds of diseases. Now, friends, I personally worked on 2-3 different kind of biological control agent, and the major one is trichoderma, where we made a nice dent. We have large number of patents on trichoderma, their formulation, and technology also have been transferred to many industries. They are manufacturing trichoderma-based biofungicide, selling to farmers, and I believe the product is doing well. That is why the farmers are buying our product on the market. You all know, in fact, I was very carefully listening Professor V.K. Sharma yesterday. He was my colleague, but I was in BHU. All across the globe, trichoderma has been reported, and it is reported from tropical countries, temperate countries, from all different type of agroecological niche, and therefore trichoderma is a well-known fungus today. Now, my group particularly, we have worked on two different species of trichoderma. One is Trichoderma harzianum. You can see here the green plus green growth in petitis, and if you see in the light microscopy, the conidiophores and conidia, they are dumbbell shaped. Okay, here, and these are the conidia. So this is how they look like. But if you see in the electron microscope, that is scanning electron microscopic picture, you can see here that dumbbell shaped structure, and on the tip the conidia is attached. So this is the typical characteristic of Trichoderma harzianum. You can identify by light microscope as well as using the electron microscope. Another species on which we work is Trichoderma virens. See the growth pattern in the petitis, you can get ring-like structure. The culture color is also dark green. And the conidiophores and conidia, here you get the conidia in bundles, like grapes. So in this particular case, you see, you can easily identify Trichoderma harzianum and Trichoderma virens. And the same can be seen in the large view of electron microscopy. You see on the phyllates, they are known as phyllates, and these are the conidia triangle phyllite, like grapes. So on these two different species we work. So we have number of publications and patents also on this. I will be elaborating later. See, 15, 20 years ago, when we were talking about biological control, everybody was laughing that how these tiny microbes, they will control pest and diseases. But believe me, friends, today, all across the world, people are using biological control, and this is the map which I picked it from the annual review of phytopathology of the article of Lorito. You can see all across the globe, people are using trichoderma. Now, not only trichoderma is used as a biopesticide, there are a number of microorganisms which are used as a biopesticide. So first one, the gadget, which was notified by Government of India, was on March 26, 1999. And they notified 9 microbes for the production of biopesticide. On the left panel, you can see the antagonistic fungi and bacteria, and on the right panel, entomogenous fungi and viruses. These were the 9: Bacillus, Pseudomonas, Glycolidium, and Trichoderma, and Bavaria bassiana, Metarhizium, Verticillium, Glyphosate viruses, and nuclear parasite. These were the 9 microbes. Later on, 25 microbes they were added in the gadget schedule till date. So therefore, if you wish to produce biopesticide in India, you can use only these 34 microbes. I'm extremely happy to say that the last microbe added in the gadget schedule was Piliformospora indica, which was discovered by Professor Ajit Verma. When he discovered, he was in JNU. Later on, he moved to MIT, and he spent a lot of time in MIT. And I can tell you, there are more than 300 research papers and number of patents on Piliformospora indica. Earlier, he reported that it looks like a mycorrhiza-like fungus, but later on, it has been proved that it is a endophytic microbe, it is endophytic fungi. So this is also scheduled in gadget for production of biopesticide. So friends, 34 microbes, they are the registered microbes, they can be used for production of biopesticide in India. Now, if you see the today's scenario, the microbes are controlled, are governed by central insecticide. If you are producing microbial pesticide commercially, you require to take license from Central Insecticide Board Registration Committee located in Faridabad. So, my dear friends, 970 microbial products are registered in India till date. And segregation, you can see here, fungal waste are 561, bacterial waste 233. And they are registered in 2 different categories. For the student particularly, you must know that there are 2 distinct types of registration with CIVRC. One is under section 93B, we call it provisional registration, and the other is under section 9(3), that is a permanent registration. So you will again see here that only 68 microbial pesticides are registered under permanent category, rest are registered in provisional category. Now, another important point is that from where to get the best quality microbe, and I will show you a classical slide which I showed at least in 50 different conferences, that if you are looking for a good microbial biopesticide or microbial biocontrol agent, you have to see the nature. And this particular slide I prepared when I was posted in Pantnagar, I could see that maize plant was attacked by banded blight pathogen known as Rhizoctonia solani. So on the left panel you can see the banded blight pathogen, and they are having brown color sclerotia here. And you see this is the typical symptom of banded blight on maize. Now if you see in the right panel, these sclerotia were naturally parasitized by trichoderma, and that is the one from where we picked the culture. We cultured it in the laboratory and tested against large number of sclerosial pathogen, and we could get outstanding result. And this particular strain we later on designated as NBRI1055. On this particular strain, we have a patent also. I will be showing you later. Now, how this trichoderma works. So, big story of the small bug you can see here. It is not working only as a biocontrol agent. It works as a plant growth promotion also. So, in biocontrol, activities are already defined in several research paper and review articles. But in case of resistance in the plant, it provides both type of resistance, systemic acquired resistance and induced systemic resistance. And these are the different type of compound like phenols and phenylalanine ammonia lysase. They are produced, and once the trichoderma attack the pathogen like Sclerotia ralsii or Rhizoctonia solani, they release these cellulases, chitinases, lipases, and finally they change the morphological biochemical characters in plant. Dr. Sarma was showing yesterday the accumulation of polyphenolic in the leaves and stem of the plant. Now, I have summarized here that you see, I haven't seen any chemical pesticide so far known globally which possess these type of activities. Now you see trichoderma is a creation of God. I always tell it is a gift of God to humanity, and therefore you see here vidicidal property, bactericidal property, nematicidal property, fungicidal property, and virucidal properties are reported from different parts of the world by different scientists. Plant growth promoter I showed you, it has induced systemic acquired resistance. Then here you see the genomic sequence of 10 species of trichoderma. They are available in public domain. Metabolomics has been completely done. Proteomic completely worked out, and 3,900 US patents are granted on trichoderma. 409 products are commercially available in Indian market. That speaks volume about this particular fungus. And now you see here, I again call it is a factory of chemical. 172 types of secondary metabolites are reported from this particular fungus. They are polyketides, phenol, terpenes, and alkaloids. So friends, saying that trichoderma is a gift of God to humanity, that particular slide proves that. Okay? Now, my group at different locations, we focused on these 5 pathogens. One is Fusarium oxyspore, a very notorious pathogen caused wilt in many crops. Then Phytophthora, another notorious pathogen. And 3 most important soil-borne pathogens. One is Rhizoctonia solani, the Sclerotium ralsii, and Sclerotinia solani. Rhizoctonia I showed is the example of banded blight. Sclerotium ralsii causes color rot disease in many plants, and Sclerotinia causes white rot. And if you see the host range, you will surprise that more than 550 different crops grown all across the world they are attacked by these crucial pathogens. Why I am mentioning that they are important pathogens? Because if you see the chemical control of the pathogens, they are not successful. And therefore, our focus was that can we suppress the growth of these crucial fungi? And if we succeed, we will develop a formulation and a product which can be used for controlling these three notorious pathogens in different type of crops. Now see the symptom of these pathogens on different crop. On brinjal, it's a very, I just given one or two some examples to show you that what kind of symptom they produce and how many sclerotia, thousands and thousands of sclerotia they are produced on the surface of the plant, and once they fall on the ground, they survive in the soil for 7 to 10 years. And there is no chemical control available which can completely kill the resting bodies of these pathogens. Another important crop is opium poppy, it's alkaloid bearing plant. Narcotic crop, you see this is attacked by Scutenia sclerosiorum, and once the pathogen attack, you don't get 1 single milligram of alkaloid from this particular. One of my PhD students did work on this. Now, gladiola is a very important crop grown in Lucknow and surrounding area. Very good cut flower. It is attacked by wilt pathogen, and farmers they lose crop completely. Not only that, they are soil borne and they infect the root zone or the stem. Sometimes there are reports that sclerotium oryzae causes leaf spot disease also. They are rare, not very common. Sclerotium oryzae in general cause only collar rot because they attack the plant at the collar zone. But there are reports that some sclerotium oryzae they infect leaf also, and this is the leaf symptom. Now, once you have identified the microbe, which is potential in biocontrol, then the second question is how to mass multiply. So our group has done a lot of work on multiplication of the, earlier to our group, work. Grain-based formulations were available in the country. People were growing trichoderma, sorghum, wheat. For the first time in the world, I can say, we grew trichoderma distilled waste of aromatic grasses, and you can see the growth. These are the traditional type of trichoderma grown on grains which are edible and they are eaten by us. But the distilled waste, which is a waste, and you can produce vermicompost and you can grow even trichoderma or other microbes which are used for biological control. So see the beautiful growth here. And on this particular simple work, we got US patent in January 2003. You see, that time the patent era was just it started. You see, we started patenting our microbe and microbial products way back in 98, 99. Now, you see here, not only the distilled waste, we tried pseudostem of banana, compost, spent carb, maize meal, rice, sawdust, sorghum grain, used tea, wheat bran, and wheat. All these, different kind of agricultural residue can be used for growing trichoderma, and you can see the growth here. This is the CFU or cell colony forming unit. Shelf life for 3 months, 6 months, 9 months, 12 months, 15 months. You can see the formulation. The spores survive up to 15 months. These are the different type of treatment, seed treatment by biocontrol agent trichoderma. This is seed biopriming. Here the difference is after treatment, you have to put your seed in moist chamber for at least 24 hours so that the microbe colonizes the surface. Now see here on the left panel, these are the primed gladiolus seed and this is unprimed. So once you prime the biological control agent on the seed surface, they provide bulletproof jacket to the crop and crop is free from soil-borne diseases. Now on potato, we did priming experiment. You can see the beautiful growth of trichoderma on potato. Now this particular isolate we patented, and you can see the patent number here. You just put in the Google, you will get the detail of that. And this particular strain has been commercialized to many companies. I will be showing you later on. Now see here what was the uniqueness of this particular micro. One, it has got a wonderful mycoparasite activity. You can see here, Rajectonia solani is completely colonized by the trichoderma. The thinner one is trichoderma, and the thicker one is Rajectonia solani hyphae. They have been colonized. Another thing is that we, for the first time, reported that trichoderma nematode control also is possible. So these nematodes are Peladovan incognita is just coiling the trichoderma is coiling this, and plant growth promotion. So 3-in-1 activity for the first time reported, and that was the reason we got patent, US patent on trichoderma. And yesterday's question to Dr. Sharma, do trichoderma induce root proliferation? Yes, they induce, and you can see the difference in maize. You can see here in chickpea and in cauliflower. So the root biomass, ours is the only trichoderma strain where we have done a lot of field trial, and the companies, they got the level claim of 8 different kind of crops with their pathogens named listed here. Groundnut, cauliflower, cotton, sugarcane, chickpea, maize, sunflower, and cumin. These are the crops we used against these diseases, and they are very, very effective. Now you can see the field experiment here, because when we transfer the first technology, the company said we have to see the experiment in field, and once we are convinced, then only we will take technology. See, this experiment are done in Pantanagar University. The sick plot is known, and you can see here on the right one, treated one, untreated, treated, untreated. Untreated means complete wilting of the plant, and this work also published in Journal of Agriculture. This is the experiment on plant growth promotion. You can see, since the time is limited, I will rush through sunflower. You can see the yield increase is about 32%. In glandulus, this you see here, sometimes the severity is so much that there is no germination. See in the lower plant growth promotion in glandulus. See, all are plant growth promotion by trichoderma. See here, we did this experiment in the NBRI, tuberose and chrysanthemum. Now here you see, we commercialize, these are the 4 patents on trichoderma. They all are US patented. And we transferred this technology to 5 different companies. They are listed here. And our product, first product was launched in 2004 in the name of Sardarico Green Biofungicide. Trichoderma harginum, NBRI 1055 is this strain. This is US patented. And you can see the logo of NBRI here. So this technology was transferred to GSFC Barodara, and the license fee that time in 2002 was 1 crore. Same technology was transferred to another company, Balaji Crop Care Private Limited, Hyderabad. Again, you will see the logo of NBRI in the top. Government of Uttar Pradesh also took our technology, and they launched UP Derma, and these are the 9 labs available present in the UP. They are manufacturing trichoderma based product in different districts of UP. Now, the most important, friends, which is very, very relevant for this particular workshop and program is that we grew for the first time trichoderma on cow dung. Cow dung is a waste, and see here, it's a PCT. We have a global patent on this, so you can download the whole patent here. Now see here the luxuriant growth of trichoderma on cow dung. So no need of fermenter, no need of bioreactor, no need of shaker. So as I told you, God has given trichoderma to you. It's a creation of God, and cow dung or any type of dung is available. You can grow trichoderma. The technology is available with us. So anyone interested can write to me. I will send you the document pertaining to production of trichoderma on cow dung at the farm site. You don't require any laboratory for this. Now, vermicomposting also we did using floral offerings, and we got only recently a Indian patent. This is the patent, and it was granted on 24th of January last year. And here you can see the floral offerings are converted into trichoderma vermicompost, and then we added trichoderma and many agriculturally important microbes. So these are all societal work. We did it with the interest that the farmers can use your technology without taking license. Now here mass multiplication using compost, popularization of technology using the village Pradhan and all. So we got a very good project from RKVY. So this is how we popularize this technology among the rural community. And so far we have trained more than 10,000 farmers in 4 different districts of UP on production of trichoderma on cow dung and cow dung cakes. Now this one is the final slide you can see here. In my childhood, I learned that there is a story of a turtle and rabbit. Rabbit is very fast. It is a chemical pesticide, and trichoderma and other biological control agent, biopesticide, they work like turtle. So who is the winner here? You all know that he is the winner because though it is slow in action, but slow and steady, environment friendly, safe for soil and plant and human health. Long-term benefits are reported by many workers. And in the beginning, I told you, chemical pesticide, they are fast but harmful. They harm environment, kill helpful microorganisms, cause soil and water pollution, and short-term gain and long-term loss is there. So friends, my submission to you is try to use biological control agent for managing different kind of pests and diseases, and there are number of products which are there which can completely control the diseases by chemical pesticide, of course, but biological is also a very good answer. And in the conclusion, simply I want to say that promoting the use of beneficial microbe, particularly trichoderma, for biocontrol as a component of IPM to manage exclusive pathogen has emerged as one of the best management practice till date. As I told you, for exclusive pathogen, there is no chemical. Commercialization of superior quality biopesticide has to be encouraged for greater benefits to the farmers as well as for improving the soil health. So this is all I wanted to share, friends. Now, so just open for question or discussion. Thank you.
Thank you very much, sir, for an excellent presentation. So before opening the floor for our participants, I myself have to ask one question from you, sir.
Sure.
If you permit. Okay, sir. I want to ask, can we use trichoderma against this root rot of apple?
Yes.
And sir, what would be the best time for the application?
See, these are the tree plants, So from the beginning only you have to take care. Like while planting,
Yes, sir.
this trichoderma enriched compost have to be added in the pits, then you plant. So they will survive, they will multiply in the pit for lifetime.
Okay, sir.
So for the plantation crop, not only apple, for the plantation crop, it is recommended that before plantation in the pits, we must use trichoderma enriched compost.
Yes, sir. Sir, do it go well with mycorrhiza? Can we mix mycorrhiza with this trichoderma?
See, there are conflicting reports. Some of the mycorrhizal strains, they are not compatible to trichoderma.
Okay.
But some they are compatible. So you have to check the compatibility. If they are compatible, certainly you can use trichoderma and PA mycorrhizal.
Okay, sir. Thank you very much. Now I am opening this session for the audience. If anybody want to ask any question, please raise your hand, or you can just write your question in the comment box.
Right. Thank you, Nikhil. I think Dr. Enrico is there from Italy.
Sir, please go ahead. Dr. Enrico, please go ahead.
From Italy?
From Italy, yeah.
Yeah, yesterday I heard him. Very nice lecture.
He is very constant and he has spent several
He
years.
has done a lot of work in developing country like India and Africa.
Certainly.
Thank you.
That is the beauty here.
I have 2 questions.
Yes, sir.
First one is those pesticides like trichoderma, are they populations that are different from a place to another. I mean, a country like India, in northern part of India, you may have a population of trichoderma that cannot be used or cannot be effective in the southern part. Populations differ from place. The effect of populations may differ from place to another. Is any evidence of that for those pesticides, biological pesticides, or they can be used everywhere in a country? And second point is any news from use of the example of rabbit in one of your slides brought me this point. Degradation of chemical pesticides.
Yeah.
Any effect or any experience of microorganisms in degradation of pesticides and in recovery in soil activity?
So I will answer your
you.
second question first, sir. Large number of publications are there from India, particularly one institute I'm referring. There is a Bureau on Insect in Bangalore, India. They have worked on pesticide degradation using trichoderma. This is one. Likewise, there is a report from Bhava Atomic Research Center by Prasun Mukherjee. He's a very good worker, published nice papers in good journal, that yes, trichoderma can degrade many commercial fungicides available and used in crop protection in the country. Regarding the first question, sir, I will just tell you, trichoderma are the biological, they are not the magic bullets. They cannot be used across the country. Say, if you have a temperate zone, you require a different type of trichoderma. If we have a tropical zone, we require different type of trichoderma. And we, for the first time, patented a strain of trichoderma that is thermotolerant. It grows up to 42 degree and it can survive up to 55 degree for 2 hours. And that strain is popular in the country all across, like particularly in southern India, because where the temperature sometimes touches 48 to 50 degrees. So for cold countries like yours, you require different kind of micro, and for tropical country, we require different. Unfortunate part is that since they are regulated by regulatory body in our country, in your country also they are regulated. So most of the companies, what they are doing, they are taking technology from a single institute. Single strain is popularized all across the country, and that is the region of the biggest failure region is that they are not working in the farmers field. So we have to develop local strain, particularly for different zones. Then only the biological control success story can be seen. Otherwise, using single strain. And in India, sir, I will tell you, there is institute like Indian Institute of Horticulture Science, Bangalore. They commercialize this technology, trichoderma herbaceum technology. It has been taken by 550 companies in our country. Meaning thereby 550 companies are manufacturing based on single strain of trichoderma. And that is sold all across the country. And that is the reason trichoderma and other microbials, they are not working in different agroecological. So your question is so valid that we have to develop region-specific strain for biological control.
Sir, I have one question. Apart from its fungicidal effect, whether it can be used, trichoderma can be used as a bioherbicide. There are some reports.
Yes.
Have you tried as bioherbicide?
I know, I haven't tried, but I listed in one of my slides that trichoderma is reported as a herbicide also. And that reference is from Jabalpur. There is Institute on Wheat Science, ICR Institute. They work in wheat crop. So once you use trichoderma in wheat crop, there is no weed. So there are 2-3 papers from that institute. Other institution also we have a paper. But large scale application of trichoderma is a VD side I haven't seen so far.
Okay, thank you. Audience, do you have any questions from the audience side?
Yeah, let me make
Yeah,
some
okay,
important
please.
discussion with my best senior professor, Dr. Singh. Sir, we are very delighted today. You delivered a nice talk. And certainly, I will use two slogans over here, who is who and how diverse is diverse. You know much better, sir. And your trichoderma certainly growing at high temperature. If it grows at psychrophilic or the psychrotolerant conditions, then we shall call it as a magic fungus, if you agree with this. Thank you, sir.
True. And that is the reason 5 companies, they paid 1 CR for our technology, whereas the ICR technology is available in 1 lakh.
Okay.
And these are barcoded microbes.
Okay.
And we have a system of catching those, companies who are using our
Right. Right. So thanks to this magic fungus and to you also. Thank you.
Thank you.
Thank you.
Yeah, participant, I expect questions from students,
Certainly. There is vacant, chat box is vacant over here. Anybody?
I didn't, show the data, I just showed you the success story only.
Certainly.
All
Participants,
publications are there, we have around 40 publications on microbes.
Thank you very much, sir, for patiently addressing our questions and providing such valuable insights. We deeply appreciated your time and
Nikhil,
contribution. Yes, sir.
there is a one chat box, the
Okay,
one
okay.
questions from participants. Please deal with it.
Okay. Yes,
Yes,
sir.
Raj.
Please go ahead.
You read at chat box. Sir, the question is,
Yes, sir.
question is, what is alternative to distillate waste for mass production?
You see, I showed one slide. The paper is published in Bioresource Technology. Any agriculture residue
Right.
which contains cellulose, trichoderma can be grown on that. Okay? And the best example I showed you, a PCT, a global patent. We call it PCT, Patent Cooperation Treaty. That cow dung we have used as a medium for growing trichoderma. And on this particular work, I was awarded by Honorable Prime Minister of India in 2011, Dr. Manmohan Singh, as a Rural Technology Award. That award carries rupees 10 lakhs cash and a citation. So that is why in the beginning I said, I never did molecular biology in my career, but I got so many awards based on my simple work, which I transferred to farmer and the industries.
Right. There is another question comes from participants, Sarvesh
Yes, sir.
Singh.
So
And
the question is trichoderma effective against wilt of guava particularly?
I haven't worked on guava, but trichoderma for wilt management is reported from different part of the country, including from CISL, Dr. A.K. Mishra. You can go through the paper of Dr. Mishra and Dr. Gupta. They have worked on trichoderma managing guava wilt. Okay, so trichoderma, as I said, it list is so big. Around 37 different kind of fungal pathogen they are controlled by trichoderma. But depends on at what particular stage you have used. Once the wilt is there in the plant, nobody can save that plant. Nobody on earth can save the plant because the pathogen has entered in the xylem system. So in the beginning, I told you, for the fruit crop particularly, you have to use this trichoderma enriched manure, whether it is a compost or vermicompost or any agricultural residue waste, then only wilt management will be there. But for wilt management, I will tell you, there is another fungus. It was discovered by Dr. Vinita Sen from IARI. Kali Sena is the product available. Kali Sena. Okay. Kali Sena is the best for controlling wilt of guava. And that is Aspergillus species. It doesn't produce aflatoxin. So aflatoxin minus strain of this particular fungus has been discovered by Dr. Vinita Sen. long ago, and the company they commercialize and the product is available in Indian market as a Kali Sena.
Okay, okay, sir. Thank you so much. Dr. Nikhil, please proceed with the next talk. Thank you so much, sir. Really for your valuable lecture. We are all very grateful.
Thank you, Jeff. Thank you.
Thank you, sir.
Go to the next one, otherwise you will be delayed.
Okay. See you, sir, somewhere in Amity. Visit us.
Sure, sir. Sure.
Okay. Thank you, sir.
Thank you.
Okay, sir. Thank you very much. So, very good morning to all. It's time to introduce our next guest. He is Dr. Siddharth Tiwari, sir, our second speaker for the session 1 of day 2. Dr. Tiwari is a distinguished plant biotechnologist whose research focuses on crop biofortification, metabolic engineering, and genome editing technologies. His work aimed at developing nutritionally enriched crops such as banana and wheat to address micronutrient deficiencies and strengthen food and nutritional security. His research utilizes advanced tools including CRISPR-Cas, genome editing, RNA interference, genomics, metabolomics, and bioinformatics for sustainable crop improvement. We are delighted to have such an eminent scientist with us today. Sir, the platform is all yours. Please,
Thank you.
sir.
Thank you so much, Dr. Nikhil, for introduction about me and my work. And before starting this presentation, I would like to say my sincere thanks to Professor Chaudhary, Dr. Karthikeyan for giving me an opportunity. So I am just going to start that my slides. I hope you are able to see this. Please let me confirm if you are
Yes,
able
sir,
to
it
see
is
this.
visible.
Right.
Yes, sir, it is visible. Please go ahead.
Right. Yeah. So thank you so much to the organizer for giving me an opportunity. I am going to present my research work in this particular conference or meeting related to precision genome editing, metabolic engineering, and biofortification for the production of climate smart staple crops. So in this particular presentation, I am trying to just summarize about my research work on banana, what nutrition I am trying to improve by using genome editing technology. So before going to start this presentation, I would like to just briefly introduce about my organization. It's known as the National Agri-Food and Biomanufacturing Institute, in short NABI, located in Mohali, Punjab. And the vision of my institute is very clear: to provide food and nutritional security to all through agri-food biotechnology and biomanufacturing research and innovations. So considering that mandates of my institute, I am working on biotechnological approaches for biofortification of crop plants. Biofortification means to enhance the nutrition quality of plants by using two approaches. One is the gain of function. That approach is also known as a transgenic or cisgenic or overexpression of gene. The second is the loss of function, where we can use RNAi approach, or most recently we are trying to implement CRISPR-based editing in plants. So in this presentation, I would like to briefly let you know about that how we can do the genetic modification in crop plants. There are four major approaches, and most commonly used is a conventional plant breeding approach. You know about this. In this particular methods or approach, what we have to do, we have to know about that one of the varieties, related varieties that is known for disease resistance. This is just an example. And second one is that allied variety, those are disease susceptible but high commercial value. So what we can do, we can go to cross these two varieties, and after that subsequent back crossing or subsequent selfing, we can get that the trait of that resistance has been transferred to the allied variety. And after, maybe depends upon the crop plants, it takes nearly 8 to 10 years to reach the product developmental stage where that trait has been transferred to the allied variety. But there are certain drawbacks in this methods, because it is limited to the related varieties. You can't able to do that crossing between wheat to rice. You have to be select the wheat varieties. Those are having these desirable characters can be transferred to the other varieties. And also that undesirable traits can also be transferred during the crossing. So this is that about the conventional plant breeding. The second one is about that plant mutation breeding. That is basically a random mutations can be created into the crop plants by gamma radiations or chemical mutagenesis. So you will generate that number of plants or populations, and based on the screening, you can identify that certain desirable mutations has been happened in the crop plants that might show that your what we can say traits that you need. So based on that, you can get that plants. But this is also a limitations like that undesirable mutations can happen to the crop plants, and the plants may not be as per the what we can say precisely mutate as we required for a particular traits. Now we can come about the third methods of genetic modification, plant transgenic breeding or plant transgenic crops development or GMOs. In this case, again, you can go to identify the gene that is responsible for the desirable traits. Like in this particular case, we are discussing about the disease resistance. So that gene we can take from any living organism, including like giant animals like we can say whale or small microbes. That gene we should know about that importance. So that gene we can go to clone in the vector or plant expression vectors, and by using the various methods of transformation we can transfer that gene into the allied varieties, and subsequently by following the tissue culture methods we can generate the plants. But this method also has some limitations, like when you are going to perform transformation of that particular gene or transgene, you will get that not precise mutation, it's a random mutations, so that for random integration in the genome, so that what happened in this case, that some of the important genes may be disrupted, so that you will get some undesirable effects of this kind of integration in the genome because it's a random integration. But fourth and very recent advancement is about genome editing. This is basically a very precise mutations you can create in the genome in a particular gene. So that is the beauty. That's why people are nowadays preferring to go for genome editing, because you are not going to disturb the function of other important genes. You can go in the targeted manners. So my research is completely focused in these two modern technologies. One is transgenics and another is the genome editing. So how you will generate that genetically engineered plants by using transgenics or genome editing? These are the certain important steps started from optimization of protocol of regeneration, that you can take the explants and perform plant tissue culture optimizations for that particular crop plants. Then you have to be identify the target genes. Then based on the identification of target genes by using different bioinformatics tools, you will go to perform that designing of vectors, that binary vectors or plant expression vectors can be used to transformation of the plants, and that optimization of transformation methods is also important on that particular crop plants. And by optimizing the methods as for tissue culture as well as transformation, you will generate the transgenic events, and that transgenic events or genetically engineering events you will go to confirm by using various techniques like bioinformatics, molecular methods like PCR or Southern blotting, and agronomical and biochemical performance. So these two steps are very, very critical in case of genome editing, that selection of target genes where bioinformatics plays major role. There are several softwares and tools are available that can be used to design that your guide RNA or target genes for that targeting through genome editing. And in the end, when you have generated the events, you have to be screened that mutation has happened or not. Again, that bioinformatics plays major role. So I am having experience more than 24 years in all these technologies and approaches that I am going to be discussing in brief. During my PhD, I did work on development of genetically engineered groundnut or peanut for protection against to the pest known as a tobacco cutworm or Sphaerophthora litura. In this case, you can see that groundnut seeds have been used for that generation of preparation of explant source, that is a cotyledon or de-embryonated cotyledon, where that embryonic portion has been removed. And this de-embryonated cotyledons can be able to generate thousands of shoots from the single explant. That the optimization protocol have already been published in Plant Cell Tissue and Organ Culture in 2008. So this is that optimized protocol we implemented for the transformation of CRY1 EC gene. That CRY1 EC gene is similar to that BT cotton where that we can get the protection against to a particular insect that is known as a Spinotra littora. You can see in this particular slides, or in this particular portion of slides, you can see that non-transgenic leaves, when insect has been released, the leaves completely chew by the insects. But in case of transgenic leaves, insects are not able to that ingest or digest or just use to chew these leaves and not able to survive. So this is the gene that we expressed by using the wound induced promoter. So that insect when attacked, only that gene will expressed, otherwise that gene will not work anything. So this is that designing of what we can say gene construct. When that insect is going to attack, only that gene will start expressing, otherwise gene will not do any work. So there is no unnecessary load into the plants to express CRISPR EC protein without the requirements. So these kind of work that we have published and perform and develop that particular varieties of groundnut. And for this work, I got that Indian Science Congress Young Scientist Award in 2010 in the section of plant science by our Honorable President and Bharat Ratna Dr. Abul Kalam Azad. Subsequently, I also used the plants as a production bioreactors for the vaccine production. In this case, that you know that plants, if you can selectively use like seeds, like groundnut seeds, if we can say, it is known for high protein storage, and that protein can be stored for a long time. There is no need of refrigerations. So if we can target vaccine candidates into the seeds of groundnut, there are certain advantages like easy transportations. You can store that particular materials in that room temperature. There's no cold chain is required. That is usually required for that commercially available vaccines through the animal cell line based system. So in this case, I approached and tried to use that groundnut seeds for the production of vaccine against to the rabies. In this case, I have used rabies glycoprotein. That is the similar structure of that you know about COVID. That outer surface of this is known as a S protein or as spike protein, that is a riboglycoprotein RGP, and that we have fused along with that cholera toxin B subunit. Cholera toxins B subunit is again is a known as that adjuvant, and if we fused, then it will start absorbing in the mouth itself. It is not going to the intestine. So that's why with that fusion we expressed by using the legumin promoter in a seed specific manner, so that that complete fusion of vaccines can be expressed in the seeds of groundnut, not in other parts. So that work we had performed for the rabies glycoprotein fusion with cholera toxin B subunit to get protection against to the rabies as well as cholera. And we estimated in this particular slides, you can see if you can express that protein in the seeds of groundnut and the expression is only 1% of total seed protein, you are able to provide immunization more than 400 million pupils from 1 acre of crops. So this is just an estimation that we had published in Biotechnology Advances, and that product of that vaccine product has been developed during my PhD and transferred this technology in Hyderabad to one company. Those are making that commercially that cell line based rabies vaccines. So in this particular work, I would also mention that why this work is important, because very recently in 2020 that we know about that COVID has came out, and for that Canada is the first country to use that plants for the production of COVID-19 vaccine, and that vaccine they have used for the immunizations of adult people those are having more than under the age of 65 years. So this is the advantage, and you can see that revolution by using plants as a production of vaccines, and it has huge advantages as compared to the normal commercially available vaccines. For this particular work, I got another Young Scientist Award in 2010 that is known as the BioAsia Young Scientist Award. So now I am coming about that topic that is known as genome editing, CRISPR-Cas based genome editing. In this case, that is the type of one of the genetically engineering approach where we can go to disrupt the function of genes by using insertion, deletion, modification, or replacement in a very precise manner in any living organism, including prokaryotes or eukaryotes. For this particular discovery that in the field of chemistry in 2020, two lady scientists got what we can say that this Nobel Prize in the field of chemistry that is known as Emmanuel Carpentier and Jennifer Doudna. So how it work? I discussed briefly about that chemical mutagenesis and physical mutagenesis. Those are able to generate random mutations in the plant system. But in that case of CRISPR-based mutations, where Cas protein can be used to implement along with the guide RNA to identify the precise mutations in the plants or any living organism, you can see in this particular examples that original gene that is having that DNA can go to transcribe into the messenger RNA, and messenger RNA can translate it into the amino acids or protein. And this is in the normal case. But when we are implementing that CRISPR-based editing, where that in this particular nucleotides, guanine has been replaced or deleted, so what happened in the mutated lines, you can see that the frame shift mutation has been happened, so that final protein or end product will not be formed due to the stop codon generation in between. So this is that precise mutations where we can be stop the function on a particular gene in the genome of any living organism. So how it evolved, basically it is evolved from the prokaryotic systems, that some of the bacteria has having that innate immunity against to the viruses or bacteriophages. In this particular slides, you can see this is the bacteriophage or virus is going to be infected to the prokaryotic cells or that bacterial cells. And when it is going to be transferred their genetic material or DNA, it is going to be integrated in the genome, that CRISPR locus of the bacterial systems, and it is transcribed into the pre-CRISPR RNA. And this pre-CRISPR RNA hairpin loop kind of structure will be recognized by the Cas protein. Or if that same virus is going to be attacked again to the prokaryotic cells, what happened? It make that this kind of formations, that is a and this formation will be recognized by Cas protein to just chop it out or degrade this DNA, so that virus is not able to infect bacterial cells again, because it got that innate immunity against to the viruses. The same approach has been evolved and developed to use that CRISPR-based mechanism in the what we can say creation of mutations in any living organism, including plants, humans, or cell lines. So in case of plants, these are the pipelines for generation of events. First, you have to be select the gene that you would like to get mutations, or that we can say silencing of that gene, or using the some of the softwares or tools like breaking case, you can design the guide RNA. That is nearly 20 base pair of sequences. That is a complementary strand of that particular gene. And that gene or is the 20 base pair sequence can be cloned into the plant expression vector by using CRISPR vector machinery, and then can go to be transformed by using different methods of transformation like agrobacterial mediated transformation, particle bombardment, nanoparticle, or what we can say magnetifications. So by these approaches, we can integrate or transfer that particular guide RNAs by CRISPR vector mechanism into the plant system, and then plants can be regenerated in that regeneration media by using tissue culture approaches. Subsequently, plant will be survived, acclimatized, and we can go to perform mutation screening by using sequencing that is most commonly used like Sanger sequencing. And there are other methods like PCR genotyping, T7 endonuclease can also be used to confirm, okay, there is a editing or not in that your plants that have been generated. So this is that standard pipelines can be used for the generation of events. In the very advancement about this technology, that government of India has been relaxed if all kind of regulations that we usually followed for transgenic event generation. In case of that, plants can be mutated by using CRISPR-based approach. But important is that we have to be proved there should not be any integration of new gene in the genome of plants. Only you have to be create mutations or disturb the function of a particular gene. So this categories of SDN1, SDN2 has been exempted by Government of India in March 2022. That's why in India you can see there are several projects have been coming out for the development of that genome-edited crop plants. So this is the list in the worldwide. You can see this is the website. If you like, you can get this tracker information of all the genome-edited crop plants. Those have very fastly reached into the commercial stage as compared to the transgenics or other technology. There are several products of various crop plants have been already been approved by the different countries for and already released in the markets for commercial cultivations. So in India, also not behind to this technology, you can see that we have also released the first rice variety that has been genome edited for environmental release, not commercial, but environmental release. And in the last year only, that two varieties have been developed by two institutions. One is that IARI, and another is that Indian Institute of Rice Research in Hyderabad. These two institutions have been developed the rice varieties. Those are having those are showing that high agronomical improved characteristics as well as drought and salt resistance in the rice. So these varieties are in the advanced stage, and we are hoping that very soon it will be available to the farmers for commercial cultivations in India. So in general, if you can see that genome editing applications for plants has no limitations, you can go to generate the plants for biotic, abiotic stress tolerance. You can enhance the micronutrient qualities. You can develop the seedless varieties and also reduce the anti-nutrient factors. But it is also limited by some of the limitations. Like first, we have to know the whole genome sequence of the crop plants that you would like to go for genome editing. You have to be carefully select the softwares for designing of guide RNAs, so there should not be any off-target impacts. You have to be optimize the protocol of tissue culture, and finally you can go to deliver the plants for this using CRISPR vectors. The selection of CRISPR vectors is also important. So these are some of the challenges. If you can take care about designing of that CRISPR based experiments in your lab for that plants or any kind of living organism, you have to be take care about these kind of limitations. So in general, we have recently published that one of the very good review article in plant molecular biology. If anybody is interested, that can read about how we can be generate transgene-free edited crop plants for that through using genome editing and also their regulations in various countries. So in my lab, we are completely focusing on biofortification of banana for enhance of nutrition quality, and these nutrition is provitamin A, ascorbic acids, and iron. And we are using tools genomating that I told about, that generation of silent mutant lines. Those are having silencing of a particular gene. It's known as the knockout events. We also try to use that implementation of new gene in a desirable locus in the plant system, that is known as a knock-in, and also transgenic approach. So we are also working on that optimization of that methods or tools for transgene free genome editing using a ribonucleoprotein complex that is very commonly used in that laboratory. We are also trying to implement developmental regulators. Those are the transcription factors responsible for the tissue culture embryogenicity or organogenicity. We are also explore those kind of transcription factors to generate transgene free genome editing and viral vectors. So I'm not going about that advancements or details about this, but these are the methods that we are using, and it is very recently very commonly used in many laboratories to generate transient-free events. So why we have chosen banana for biofortification? Because we know that banana is India is the largest producer of banana, and more than 130 countries in the world are cultivating banana and contributing. India is contributing around 26% of the world banana productions, and the fruit is available throughout the years at the reasonable price. And most importantly, it is the choicest and suitable fruits for all age groups, including infants, adults, and old people. But you know that a banana is a basically genetically sterile. The genome is in triploid, so there is no possibility to go for breeding kind of approach. So only biotechnological can provide the solutions to improve that at genetic level of banana. That's why we have chosen banana for nutritional improvement by using genome editing approach. Vitamin A deficiency that we would like to target by enhancing that provitamin A content in fruit of banana. So before that, you should know about that vitamin A deficiency is the serious health problem in the world, specifically in the developing countries. And it is reported that more than 6.5 lakh children die just because they are not getting vitamin A sufficiently in their diet. And another 5 lakh children go permanently blind. So vitamin A deficiency is responsible for mortality, blindness, night blindness, impaired immune system development, and impaired brain development. In case of India, you can see this data we have taken from that ICMR NIN Hyderabad. It is reporting that nearly 57% of children, those are having less than 5 years of age, are suffering with vitamin A deficiency. And it is also reported death of more than 3 lakhs per annum. So that's why we have chosen to enhance that provitamin A. And how that you can get the vitamin from that food sources? There are two ways you can. If you are non-vegetarian, you can get from animal sources, and if you are vegetarian, you can go for plants. In non-vegetarian, they can directly get in the form of retinol for vitamin A. But in case of plants, you have to get pigmented carotenoids. Those are alpha carotene, beta carotene, and gamma carotene. And these are pigmented carotenoids known as a provitamin A. That that beta carotene has highest activity to convert into the vitamin A in our body by the enzymatic activities. And most of the Indian populations, that is 30% around that, is vegetarian. That's why we have chosen that plant source to enhance that nutrition quality, particular vitamin A in plants. So how much that vitamin A we have to get? That is another study, background study that we did. Again, we got that information from ICMR. It is reported that we have to get 1000 microgram vitamin A per day in our diet. And the conversion ratio of beta carotene to vitamin A is 1:6. That means if you are getting 6 molecules of beta carotene, it is going to be converted into the 1 molecules of vitamin A. Based on this conversion ratio, we have screened the Indian banana germplasm and find out most commonly cultivated variety Grand 9 is containing only 400 to 500 microgram per 100 gram of weight of fruit pulp weight. So this is that banana pictures. You can see this is the eatable portion of pulp. It is having only 400 to 500 microgram of that beta carotene. So based on this estimation, we find we have calculated that we have to be get increase nearly 15-fold content of beta carotene to the fruit pulp to get 100% RDA that can get from the single banana. So based on that, again, we have done some background study about the understanding of carotenoids biosynthesis pathway. It is started from MVP pathway, where one of the rate limiting enzymatic steps regulated by DXS gene, deoxyxylose 5-phosphate synthase. This gene is going to be provide substrate flux into the carotenoids pathway. In the carotenoids pathway, it is started from geranyl diphosphate, GGPP. It is converted into the phytine by the activity of phytine synthase. And phytine is going to be down into the synthesis of lycopene by the activities of various enzymes like PDS, ZDS, so, and this lycopene is diverted into the two direction. One is the synthesis of alpha carotene or lutein with the activity of lycopene epsilon cyclase. But lycopene beta cyclase is responsible for the synthesis of beta carotene, and this beta carotene is going to be degraded by the activity of VCH and CCD. So based on this complete known pathway, we designed our approach for the gain of function, that is overexpression of genes. In this case, that upper pathway, DXS and PSY, we have considered for the overexpression so that we can go for gain of function approach and loss of function by using CRISPR-Cas approach. We have first selected PDS gene, that I will let you know why we have selected this. If we can target this gene, we can just use as a proof of concept. And subsequently lycopene epsilon cyclase, that is diverting lycopene in other pathways. If we can silence or edit, that lycopene go to synthesize beta carotene. And that in the downstream of beta carotene, BCH and CCD is again going to be targeted by CRISPR approach so that beta carotene will be stabilized. So we have designed our approaches by using or considering this pathway. Regarding genome editing, we did work on that PDS gene, lycopene epsilon cyclase editing, BCH editing, and CCD4 editing. That work has already been published in different journals in different time frame. You can see that PDS gene we edited as a proof of concept, and that work was published in Functional Integrative Genomes in 2018. Then again, lycopene epsilon cyclase we edited, and that work was published in Metabolic Engineering. Then CCD4, that is responsible for beta carotene degradation, has been published in Journal of Experimental Botany, and BCH editing has been recently published in Transgenic Research. We also published some of the very good review articles in various time frame, started in 2019 to 2025. If somebody is really interested to go more details about that, how genome editing is important and how that can be implemented to improvement of crop plants at genetic level, you can go and read these kind of review articles. Might be you will get more ideas and understanding about the genome editing. So about the first target, as I told, is a PDS. This PDS is basically just behind to that phytine synthase, and phytine is going to be what we can say converted into the lycopene by the activity of PDS, that is phytine desaturase. If you edit or silence this gene, that carotenoids will not going to be synthesized. Also, this gene is responsible for the chlorophyll biosynthesis. If you edit this gene, you will get some albino phenotypes. Albino means yellow kind of leaves. There is no chlorophyll biosynthesis. Based on that, you will judge, okay, that editing is happening or not. So following that approach, we designed the guide RNA and perform that transformation in that embryogenic cell suspension culture of banana cells. You can see that albino phenotypes has been noted during the tissue culture stage, that white color of albinos. But control embryos, those are not having that editing, is showing that complete normal phenotypes. So that plant subsequently generated through the mutated lens will show that albino phenotypes. So after confirming that editing is happening or not, we had performed that sequencing, and the sequencing have been shown that generation of frames with mutations or stop codon generations, so that PDS gene is not going to be synthesize the protein. Subsequently, you can see that all that edited lines have been shown low carotenoid depositions compared to the control, also the low depositions of chlorophyll content as compared to the control. So this is the proof of concept that we have first demonstrated in the world that CRISPR-based genome editing is possible in banana. And that work was published in Functional and Integrative Genomics in 2018. Subsequently, we identified the target lycopene epsilon cyclase. As I told, this is diverting the lycopene resources to the other sites, but our target molecules is beta carotene. So we designed lycopene epsilon cyclase. The construct has been designed. The tissue culture approach has been followed to generation of plants. The mutant lines have been confirmed by the sequencing, and also T7 endonuclease acid that we had performed. And then you can see that around 6-fold content of beta carotene has been increased in the edited lines for that lycopene cyclase editing. And also you can see that dark yellow color phenotypes in the edited banana due to the high depositions of carotenoids. So we also perform some agromorphological data analysis in the edited lines and control lines in that NetHouse based study. And it shows that the plants, those are having a high content of beta carotene, that is 6-fold high, are showing that comparable phenotypes to the control. And that work was published in Metabolic Engineering 2020. And that was the first report in banana to improve any nutritional traits by using genome editing. The other target is CCD. That as I told, that along with BCH, CCDs is also responsible for the degradation of beta carotene. So we have designed that constructs for the CCD4 gene, that banana, for the targeting in the Rastali genome, that is the protoplast-based screening. We had first confirmed that editing is happening or not in the banana plants. That protoplast has been used, and then within 3 days we can make sure, okay, editing is happening in the protoplast of banana. Subsequently, we performed particle bombardment and generated thousands of banana plants and screened by sequencing and find out some of the lines those are having editing. And that lines, after confirmation, we estimated that carotenoids depositions in leaf and root tissues, those are showing 2 to 3-fold content of beta carotene depositions. And also we had performed some differential expression study of that other pathway genes. And that work was published in that Journal of Experimental Botany. That is a transgene-free editing that supports that CCD4 is responsible as a negative regulator of beta carotene in banana. So that other target is ascorbic acids. You can see that in human we have to get around 80 milligram per day vitamin C. That is ascorbic acids. And in case of screening of Indian banana germplasm, we find out two contrasting cultivars. Nandan is having around 17 milligram per 100 gram of that ascorbic acids, while that other varieties, those are very commonly grown, is Rasthali in south parts, is having only 7 milligram per 100 gram of ascorbic acids. So based on this screening, we find out that which kind of targets we can be considered for the improvement of ascorbic acid content in Rasthali. So this is the Mayox pathway. In this Mayox pathway, you can see that myoinositol resources diverted into the synthesis of ascorbic acids if we can edit that particular gene gluconokinase. This gluconokinase gene is basically diverting glucuronic acids into the cell wall biosynthesis. If we can edit this particular gene, the possibility that all resources can be go to synthesize L-ascorbic acids. For this, we have identified gluconokinase gene in banana genome. The construct for editing has been designed. We had performed the tissue culture for the generation of edited events, again confirmed by the sequencing, and that you can see that edited lines for gluconokinase are showing that improved agromorphological observations as compared to the control. With high yield, the fruit is very healthy as compared to the control, and also they are showing that high yield compared to control. So these analyses have been shown by HPLC. We had confirmed that the content has been increased 2 to 3-fold high in case of Rastali. That third target is iron biofortification. We identified after screening, we identified Rastali and Robusta contrasting cultivars, and now we are working in the mechanism how that we have already published this work for that screening. And we had developed that collaboration in Germany, where we are working on that identifying the regulators. Those are responsible for the low depositions of iron in case of Rastali. And we already identified some of the targets like IRO3 for genome editing, and work is going on in these directions. So in summary, you can see that we have generated that that genome editing and transgenic banana lines. Those are having very high amount of beta carotene depositions that is reflected by the dark yellow color. And that project has been supported by Government of India, where the Prime Minister Modi has already been visited to our collaborator in Australia. And last year only, that minister has been visited to Nabi, and he appreciated that a product has been developed and it is in the advanced stage. We have recently performed the multi-location event selection trials with this golden banana, and we are hoping in next 3 to 5 years it will be in the market. You can see that due to the high depositions of beta carotene, that banana is turned into the completely dark yellow colors just because that beta carotene content is very high in this particular lines. So we have also given that interview to that one of the interviews in that Government of India Vigyan Prasar news channel. If somebody is interested to get more information about how we can reach lab to land for development of this technology, you can get more information in this interview. So I am thankful to the research members. Those are currently working with us in various projects, as well as the previous research. Those have been well settled in India as well as in abroad. They contributed in various projects in Nabi, specifically for the development of golden banana. So we are thankful to the executive directors of Nabi for grant, core grant, as well as that BIREC, Department of Biotechnology, that our collaborators in India as well as in outsides for their supports and guidance, as well as that funding resources. So thank you so much. I'm just concluding my presentation. Thank you.
Thank you, Dr. Tiwari. So nice of you to have on this platform once more.
Thank you, sir.
And I requested you many times, kindly visit Amity offline, but you
Zarur,
never
sir,
come
zarur.
to see.
Definitely, I will try to come. Thank you so
And
much for this.
certainly, sir, you talked about superfood, I would say, if you agree with this. And the source is Nabi for distribution of this banana, or shall it come to show presence all over India in the market?
Definitely, sir, it will be come to the market by getting that approvals from Government of India. It is
Okay. So what will be the cost of this banana in the market? Sir, please unmute yourself. Sir, you are muted.
Sir, please unmute.
Sir,
Are
please
you hearing?
unmute. Yeah. Dr. Tiwari, please unmute yourself, please.
Yeah, he unmuted.
yes,
Okay.
sir. Are you able to hear me?
Yes, yes, certainly.
Yeah.
You are
Yes.
audible.
Yeah,
Sir,
please, sir.
yeah. So how many years you think that it will encroach the market for distribution?
Sir, we are expecting 3 to 5 years because it is already performed one event selection trials in multi locations in different geographical locations in India. And after getting confirmation or approval from Government of India, we can go for another trials, multi location trials. So
Okay.
after that, we are hoping it will go to the market. So maybe it will take 3 to 5 years for all that clearance.
Sir, I am ready as a voluntary clinical trial if you are ready, because my
Oh.
eyesight is very weak. Yeah.
Sure,
Okay.
sir, sure.
Thank
I
you,
will
sir.
invite you to come to see that
Right.
product in our lab or our
Thank you.
institute.
Thank you. Any questions from participants? So
Sir,
forth,
just
please.
I have a question. Like instead of tissue culture, some scientists they are doing with implanta directly agrobacter. Have you ever tried directly with implanta technique?
Yeah, as I told that there are some of the methods like where we can use the developmental regulators or viral vectors.
Yeah.
So what happened in this approach, that gene is not that is not going to be integrated in the genome, because virus is not transferring that particular gene into the plant system. It is just transiently expressed. So that approach people are trying to use, and same we have used in case of that plant transformation. What we did that we have used developmental regulators or transcription factors. Those are responsible for the organogenesis. So by using this viral vector, we have integrated that particular gene responsible for the shoot formation as well as CRISPR machinery. So this is going to be infected by the virus into the plants, and that after using that plant translational machinery or that mechanism, that gene will going to be expressed to make that de novo shoot formation. And that de novo shoot basically also having the editing, because CRISPR machinery also used along with that. So that de novo shoots we can cut from there and can grow as a normal plants. So that approach people are using in some of that advanced laboratory practices. That same we have done in case of pea, and we have that very good protocol for this in legumes crop. So we are in the advanced stage for this. Yeah.
Okay, compared to diploid, polyploid species may be having some difficulties designing because of multiple copies of the genes, etc.
Certainly.
So
Yeah,
more
the
precision is
multi-copy.
needed in gRNA designing. So can you say some suggestions, sir?
Yeah. As you are saying that if you are going to work on polyploid crops or vegetative crops, like I am working on banana,
Yeah.
are having triploid genome. In
Yes.
this case, CRISPR is advantageous because you can design the guide RNA that is common for all three
Yes.
alleles.
Yes.
This is
Yes.
the advantage in case of using CRISPR. So there are softwares are available like
Yeah.
breaking Cas or other softwares that can help to use to design that guide RNA that is
Oh.
conserved for all alleles.
Okay.
So you will
So the all
simultaneously
alleles are
target.
expected to have a similar function only. Yeah.
Right.
Oh.
That will help. Yeah.
Okay, sir. Audience, do you have any questions? Please ask. Any questions from audience?
Okay, so
Okay,
sir,
sir.
actually we are running short of time. Thank you very much, Dr.
Okay,
Tiwari.
Nikhil.
It's wonderful to listen you and going deep into the new things that are taking place in the domain of agricultural biotechnology. Thank you very much, sir.
Thank you
Thank you,
so much,
Dr.
sir.
Nikhil. Yeah,
Really
thank you,
valuable
Dr.
lecture.
Karthikeyan. Thank you,
Yeah.
Dr. Chaudhary. Thank you so much. Namaste.
So now it's my pleasure to introduce our third speaker for today's session, Dr. Debabandya Mohapatra, NAS Fellow and Head of the Automation and Plant Engineering Division at ICR, National Institute for Secondary Agriculture. Dr. Mohapatra is a highly respected agriculture engineer known for her outstanding work in grain processing, storage engineering, automation, and sensor-based technologies for agro-processing systems. She has made significant contribution towards post-harvest management, grain storage efficiency, and sustainable food processing technologies. Her achievements have been recognized through several prestigious awards, including the Jawaharlal Nehru Award and ISAE Team Award. We are privileged to have her among us. Ma'am, I respectfully invite you to share your insights with the audience. Please, ma'am.
Thank you very much. Sharing.
Thank you, madam, for coming over here. This is DK Chaudhary. Nice
Hello,
to
sir.
meet you. Yeah,
Nice meeting you.
yeah. Please.
Can you see my slides?
Yes, madam. Make it a slideshow. Make it full, ma'am. Yeah, it is visible to us. Thank you.
Thank you very much for inviting me to this FDP program. I'm really happy to. I was listening to my previous speaker, and it was really nice to see and very nice technologies coming up to be discussed in such a forum. So I would start with my work. Like though I have worked in many aspects, but recently I have been working with millets, and as for a sustainable food product, nothing is better than millets. So I'll go in two parts. I'll first is my product development, and next is storage systems for millets. So everybody knows how important is millet. It's packed in, we have more than 9 varieties of types of millets here. And if you see, they are slightly different if you look at their morphology. Where per millet and sorghum, they don't have any husk. Finger millet has a very thin husk. Kodo, foxtail, barnyard, little millet, they have hard husk. So how they offer the different functionalities also differ. Per millet and sorghum, we can directly make it flour and prepare different foods. But for others, we have to dehusk them. They are very highly nutritious, high in protein, especially foxtail millet. Then it can serve a different purpose, not only feed for fodder, and also in some medicinal, like while making tablets, also the millet starch can also be used. Especially those husked millets like your kodo millet, little millet, they have a very hard husk. So they are usually, if you can, they can be stored as such without any problem. Without any problem. But pearl millet, sorghum, they have storage problems. They are getting affected by mostly the major pests and minor pests also. So my presentation will be dealing with both the husked millet as well as unhusked millets. During the post-harvest operation, after harvesting, they usually dry it, thresh it, then clean it and store it. For millets that needs to be dehusked, they are dehusked. And those the like pearl millet and sorghum, they are directly made into flour. And packaging is also done. Packaging especially is a problem because they have a high density, especially pearl millet. They have a very high lipase activity, which makes them rancid within, say, pearl millet. In rainy season, it will go rancid within 6 days, 7 days. Then sorghum maybe 15 days. But if you store them like kodo millet, they can be stored with the husk for the years. So we need some good packaging technologies, some processing technologies to improve their shelf life, which can decrease the lipase activity and improve its functionality. So they also have some anti-nutritional factors like phytic acid, which will make complex with the metal ions. So their absorption to our body is difficult. They have tannins and phenols. They also reduce the amino acid availability. Though people say we are taking very healthy food, but if you see in the tribal region, when they are completely relying on millets, they have a very high cases of malnutrition, because whatever they eat, if they don't use the fermentation or some kind of processing, the nutrients are not going to be absorbed in their body. So that's why we require this processing. So oxalates, they also inhibit the iron and calcium absorption. Enzyme inhibitors are already there, trypsin inhibitors. So hydrocyanogens like in kodo and some other millets, hydrocyanogens are also a problem. So without processing, millets should not be consumed. So what are the different processing methods? Usually in traditional form we are using, usually soaking, sprouting is done, or boiling or fermentation is done. Mostly if you look at the traditional foods in South India, they are made in fermented form. They are consuming in fermented form. So we need to take care of their anti-nutrition first, then only we can take up the nutrients. So we are, in the last few years, millets, after the International Millet Year was celebrated, lot of companies are developing a lot of products, lot of institutes are also developing a lot of products. They have been very well adopted. But many of the products, if you see in the market, they say they are millets, but you will find hardly 10% or 20% millets in them. Getting them totally 100% millet is difficult because they don't have the binding agent like gluten. So even if you are taking millet roti like your pearl millet or say sorghum jowar roti or bajra roti, it becomes very hard and it's not very appreciated by those who are comfortable eating it, they take, but those who are comfortable taking wheat roti or atta roti, they don't appreciate taking it for a longer time. For that, we also can use different like dehulling, polishing, stratification, parboiling, flaking, blending, fortification that can be used for improving their nutritional ability. But what about their binding? How to make the roti or make it more palatable so that the general people can consume it without any problem. So we need to do some modifications in the starch or protein or fatty acids so that they're more palatable, their absorption is better in our stomach, as well as that fatty acid which makes the rancidity, they can also be reduced. So some of the fermentation techniques, like we are doing cross-linking for making a resistant starch, or we are through enzymatic hydrolysis, we are making it smaller molecules so that the complex sugar, when they break, it tastes better. When complex sugar is not absorbed on our palate, it doesn't taste good. Like your rice and wheat, they don't have, they're simple sugars. But millets have complex sugars. So complex sugars, to make them more palatable, if you can use fermentation technology or hydrolysis techniques, they are more palatable. And fermentation not only improves the taste, it also improves the amino acid availability. If you go for fermentation, like we tried fermentation, not natural fermentation, but forced fermentation using lactic acid bacteria. Now, lactic acid bacteria itself is a bacteria which has nitrogen content or say protein content. So invariably, the bacteria will improve the protein quality of the millet, which is less in millets. Except few millets, the protein content is very less. So improving to improve the amino acid profile, the mineral profile, or the digestibility and palatability, the traditional fermentation can be used, but using particular bacteria like your say probiotic bacteria or lactobacillus, some species of lactobacillus, they can improve the protein content. Also, they will improve the vitamin B. Like vitamin, especially in a vegetarian section, vitamin B12 deficiency does occur, and moreover, many people consume raw rice where the vitamin B is in the outer layer. So when on the process of dehusking and milling, we lose the vitamin B. So fermentation is one of the process, post fermentation is one of the process where we can improve the nutritional value of millets. Not only that, it will also reduce the lipase activity. So the storage life is enhanced. Besides, it will reduce the tannins, which are anti-nutritional factors or phytic acid, since their bonds are broken. So those millets have better digestibility or better absorption in our body. So we are getting the millets which all these nutrients are going to be absorbed in our body. Apart from that, you have germination or sprouting. It will also improve the vitamin B and improve the antioxidant and reduce the anti-nutrition factors, malting. Some of the thermal processing like popping, using microwave or IR heating, we can also improve the palatability of the millets and also improve the nutritional absorptibility. Microwave heating and IR heating also decreases the lipase activity, so their storage life is also increased. And microwave has also been used for disinfection or disinfestation of the grains. Similarly, high pressure processing, pulsed field, cold plasma or ozone, ultraviolet treatment, nanotechnology, they are all used for the novel technologies, they are all used for improving the texture, bioavailability of the nutrients and palatability of the nutrients, as well as making new food products. There are some of the value-added products developed from millets at CIE where I was working before. So some of the products have, like if you can see the roti, it is completely made of millet atta. No other atta is added into it. So we have used fermentation technique, which improve its extensibility and also improve its characteristics than the raw millet. Some of the probiotic millet drinks are also been developed. And we produced some of the halwa mix or kheer mix, which have higher storage life because of these processes we gone through. And millet sathi, we here in this present institute, we have developed one plant-based additive, which when added in certain quantity, say 25 gram per kg of millet atta, we can very well prepare rotis which is as good as wheat roti. it will puff completely. So that particular millet sathi we are talking about has good fiber content. So it will also improve the fiber content of the millet. So these are some of the new functional food products developed by my institutes. When I was talking about millet fermentation, how it does, scientifically you can see there is a change in the starch structure, there is a change in the protein structure. We are improving the phenolic flavonoid content and reducing the tannin content. And of course, the amylose content improves because the chain structure of the amylopectin breaks down and it produces more amylose. So amylose is more digestible form. So this is just the verification that we have carried out things scientifically. Then I'll be talking about some of the non-thermal technologies like IR heating. That also we have seen it will improve the say total phenolic content or total flavonoid content, even antioxidant activity. It also decreases the lipase activity that improves its storability. So if I compare the fermentation versus IR heating of the kodo millets, fermentation of course has a better value added product because it enhances the amino acid, fatty acid. But in some case, IR heating, of course, it reduces some of the enzymes and improves some of the quality, but not at par with fermentation. So fermentation is definitely a better practice, and many of the researchers are now coming up with the prebiotic, using millet as prebiotic. So probiotic culture and prebiotic mixture, that also makes a very good functional food for a healthy life. Now coming to next section, that is the smart storage system for food grains using AI and ML. Millets, as I already told, millets have some of the millets like pearl millet and sorghum, they definitely have this rancidity problem. And for that, how to tackle them, like if new things are coming, that we are using IoT to check whether there is a change in the moisture content, change in the temperature. So that will tell you whether that grain is going to be spoiled. So some of the, we have used temperature sensor, moisture sensor, as well as carbon dioxide sensor, because when there will be more insects, the carbon dioxide will be more. So that indicates the spoilage of the grains. So we have used some acoustic sensors also to decide whether there is any insect. Since insect will crawl and bite the grains, they will make some sound. So that kind of the acoustic sensors will catch that sound, analyze that using AI models, and then we can find out whether the insects are really how much, what could be the population of the insects that are affecting the grains. So this is one of the our students who worked on defining and developing a acoustic sensor based sensing system for insect detection. And we could found there are two that was infested with two types of insects. Each of the insect will have a different signature, sound signature. So from that we can understand whether there is a secondary signature, secondary paste or primary paste, and accordingly that we can take care what could be done. Yes, what next is, after insect detection, what we should do? We should do fumigation. We tried ozone fumigation for our grains, and it did have some like if ozone is given in a particular dosed manner, then definitely it can kill the insects. But at the same time, ozone also being a very highly oxidizing agent, it will also deteriorate some of the food like quality, like it will decrease the amino acid, it will also decrease some of the antioxidants. That's why the dose has to be proper, dose should not exceed. So whatever is dose, for that we need to have some sensors which will take care, like it will tell that yes, that is enough. So ozone treatment, it does disinfection, or if there is any microbes, that can be also killed. So all these things are fine. Disinfection, enhanced polyphenolics because the release will be better, improve dehusking in some millets we have seen, better cooking quality also we have seen. But at the same time, we have to compromise on the decrease in the amino acid and fatty acid profile, and there will be some germination percentage that will be reduced. At the same time, it could also, since it will release the fatty acid, so fatty acid, that ratio omega-3 and omega-6 fatty acid, that also improved in our case. So ozone can be used for disinfection of the grains. So achieving a safe storage, if you use IoT as well as fumigation, if you use IoT for aeration and fumigation control, we can, through AI, we can, through say, adopt the cloud storage, where if there is a say, suppose there is grain is stored in the bin and there is rain, but at the same time it is indicating that there is insect, but at the same time we cannot do aeration because it is raining. So we should, the cloud, whatever IoT based system we are using, it should decide for itself, so that the aeration and fumigation can be done automatically without destroying the grains. So that will enhance the efficiency, ensure the security and accessible to grains. Another case we have done is using VOC for detecting the insects or rancidity. So VOC is nothing but volatile organic compounds which are volatile in the normal temperature. So when we smell something, we can find that it is good or bad. So this is the same thing, it is imitating your nose. So we use metal oxide sensor, but there could be electrochemical gas sensor, infrared gas sensor, or catalytic bed sensors, photoionization detector, thermal conductivity gas sensor, optical gas sensor, and laser gas sensor. Metal oxide is very widely used, and because it is very cheap, and it gives very good result. If you check the VOC profiles during storage and per millet, you can find that even if it is not infected, you will find that the fatty acids are increasing. For different temperatures, if you store it, the storage will give you different kinds of chemicals will be generated at different stages. So that will tell that yes, my grain is right or not. My grain is going to spoil or not. So we have developed one of the student has developed one sensing system for rancidity detection. It will tell the grain is fresh. It will tell whether the grain is going to be spoiled, or it will tell the grain is totally spoiled. It based on the fatty acid profile. Like we have taken the sensor reading and measured the fatty acid simultaneously, and developed an algorithm that if this is the limit, if 8 is the factor, then grain is fresh. If 9 is there, then grain is going to be spoiled. If it is 10 or 12, then grain is totally spoiled. So that kind of algorithm we have used using two sensors, like your fatty acid acid sensors and alcohol sensors. Another sensing system was developed for in detecting the insects. Here we have used 3 sensors, that is ammonia sensor, alcohol sensor, and acid sensors. Normally in eNose, usually we use from 8 to 12 sensors to detect various gas. But here we have used minimized, because we know what kind of gases are going to be formed during the storage or during insect infestation. So by measuring the gas headspace, we found that yes, these are the components which are increasing if the infestation is more, if the rancidity is more. So accordingly, we selected the sensors and used it. So that will conclude my storage part. So when you do post-harvest engineering, you optimize all the processing parameters to develop a sustainable food through value addition, where you want to improve the nutritional quality or improve the storability, or whatever is the goal. Then you require smart storage technologies. Even if the products are produced, they need some storage techniques or packaging techniques using say nitrogen atmosphere for a packaging is a good solution for millets which are going rancid. So that will ensure our food security, and we'll be a leader in the functional food development. With that, I conclude. Before that, I need to acknowledge all my students, Sunil, Seetal, Nikhil, Silpa, Monica, and Sweta, who have worked in this area for developing for a better or sustainable food development for millets, especially for millets. Thank you very much. I think I am within the time.
Very much in the time. Thank you, madam. Very much. Dr. Nikhil, please carry on. Few minutes.
Okay, sir. Thank you very much, ma'am, for your very insightful lecture on post-harvest technology. Now I will ask from audience if anybody want to ask some question, please go ahead. First, raise your hand, and then you can also write your question in the comment box.
Professor Porchedu, do you have any question? Please ask.
I was going to ask, in fact,
Yeah, please try.
a few points.
Yeah.
The first one is the degradation of anti-nutritional factors by cooking or by processing food. That is first one. Those devices you presented for detecting gases or for preserving, in general terms, for preserving food will be costly. Would the farmers be able to afford additional expenses? Can those be done, or could those be done at village or regional level? My point is that if you try to sell the product as such, a big industry will come,
Yes.
and most of the gain will be on their side and not to the farmer's side. Our experience is that farmers are getting some 15% of the cost of food, which is very limited. All the rest, 85%, goes to other people, to industry, etc. What is your experience, and what is the expectations in India for
Yeah.
processing food?
Yeah. Thank you for your question. Yes, those devices, they are developed for grain dealers. I mean, the grain dealers, when they buy the grains from farmers or any other places, they first check it by destructive method. When there is a quality check, they do it. There are some chemical methods, but these devices quickly, within 2 minutes or 3 minutes, they will say the grain is good for purchasing or not. So that's a very quick solution for those grain dealers, or say FCI go downs, or those who are going to process their millets for them. And if they and it won't cost, these equipment hardly cost 25,000 with all accessories. So I think these grain dealers, they can definitely adopt that. It's not a very good, very high cost. As far as farmers are concerned, we are advocating hermetic storage for millets, because hermetic storage, it will produce. I could not tell much because I had to finish my topic within 20 minutes. Yes, the one you have seen with sensors, we have used hermetic storage systems. So hermetic storage system will remove all the gases, and it will produce carbon dioxide rich environment, which will not allow the insects to grow. So that way, millets can be stored safely, even if they are unhushed grains like power millet and sorghum. Other millets, for kodo and foxtail millets, we are not bothered. They can be stored as such. Especially these 2-3 millets like ragi, sorghum, and power millet, they have a problem. And we can always vacuum packaging is one of the method where the grain stores are now giving vacuum storage system for whole grains. So that can be that is also not a big issue. As far as flowers are concerned, yes, they do get rancid after 10-15 days, but with fermentation, we have seen the shelf life has increased. So for sorghum, the increase is 45 days. And for per millet, which is usually 7 to 10 days, it has increased to 22 days. So that is a big thing. And usually when in villages, they don't make the flour whole. They will just take 1 kg, 2 kg, they make it the flour and use it at the home. And this goes on. This is the usual practice. But for fermentation, they can keep it at least for 3 weeks per millet. So that's a very, and fermentation has been used by Indian or in African countries since ages. It's just slight tweak in the fermentation method that they are using. If they use some, instead of buying some very expensive culture, they can use the curd. Every house has the curd. They can just put the curd, ferment it over 6-7 hours. The fermentation time is reduced. You get the highly nutritious food, and the shelf life is more. So that way, it can go to farmers. Industry people, if they want, they can use media to improve the fermentation quality. But at home, you are using, and in fact, I have started after working on that. I have been taking only fermented millets for everybody's knowledge.
Okay,
So
ma'am, I have one more question. For example, rice is one of the crop which accumulate more arsenic. Have you
yes.
compared among the millet species about this heavy metal accumulation besides the anti-nutritional factor? And did you see any good varieties that is accumulating less level? That have you used those kind of variety? Because we are talking about anti-nutritional factor, etc. But we have to also think about this arsenic kind of heavy metals. Did you compare them?
So far, like we have not seen those kind of things, but during fermentation, yes, we found some of the heavy metals goes away. I didn't show it. I mean, in sorghum, you have seen some of the heavy metals, they are reduced during fermentation. But the water has to be
Added
I mean,
more, yeah. In rice, that is the reason we are washing again and again in general. Yeah, I think also, I think a specific variety also important, which is
I won't be able to tell you about the varieties because I'm a
no.
processing
I can understand definitely. Yeah, but I think selection in general, I mean to say.
Yes, yes.
Yeah.
Yes, there is a problem of kodo poisoning. Some of the bacteria they grow during the rooting and they go to the seeds, and after the animals take it or they die because of the high cyanogenic acid. So that's why many people have stopped especially when there is a rain. In the millets are supposed to be grown in arid, semi-arid climate, and there is a waterlogging that fungi grows, and it really there is a problem. Yes, there is a problem of kodo poison, hydrocyanogen acid.
Right. Very
Thank you,
nice,
sir.
Dr. Mahapatra. We are very delighted to have you with us today. And certainly, a lot of things came in my mind also, how to keep food safe and make it more functional. Thank you, madam. And Dr. Nikhil, I would request you please wrap up before lunch.
Thank you, Dr. Chaudhary, and thanks the team MET for giving me an opportunity to discuss with you.
Thank
Thank
you
you so much, ma'am. Thank
You're
you.
welcome. You're welcome, Dr. Mohapatra. With this, we come to the end of this session. This is session 1 of day 2. I sincerely thank all the esteemed resource persons for their enlightening presentations and all the participants for their active engagements. Okay, we will now take a break for a lunch. I kindly request all the participants to join us again for the session 2 at 2 p.m. sharply. Okay, looking forward to your active participation in the next session. Thank you very much.
I am Dr. Payal Chakraborty from MIT Institute of Organic Agriculture, MIT University, Noida. And it is my privilege to serve as the host and moderator for today's second session. On behalf of the organizing committee, I extend a heartfelt welcome to all our notable speakers and participants. Thank you for joining us. And I hope you will find today's sessions more informative, engaging, and inspiring. It is a pleasure to welcome all the distinguished resource persons, faculty members, scientists, researchers, scholars, and participants to the second day of our faculty development program. Following an engaging and enriching first session, we are delighted to continue this academic journey with the second session under Theme 2, focusing on climate-informed soil, crop, water, and post-harvest management in climate-smart agriculture. This program serves as a platform for knowledge exchange, interdisciplinary learnings, and meaningful discussions on innovative approaches to addressing contemporary agricultural challenges. We look forward to another day of insightful deliberations and fruitful interactions. To commence this session, it is my immense honors and pleasure to introduce our first prominent speaker of this session, Dr. Arun Kumar Joshi, an internationally acclaimed weed scientist, eminent plant breeder, and a leading figure in global agricultural research. Born on 2nd August 1958 in Uttar Pradesh, India, he completed his BSc in Agriculture in 1978, MSc Agriculture in 1980, and PhD in 1983 from Banaras Hindu University. Dr. Joshi served as CIMMYT Asia representative and managing director of the Borlaug Institute for South Asia from 2016 onward, playing a pivotal role in strengthening agricultural research, collaborations, and promoting climate resilient wheat production systems across South Asia. He has been closely associated with the development and dissemination of improved wheat varieties adapted to diverse agroclimatic conditions and resilient to biotic and abiotic stresses. Before joining CIMMYT leadership, Dr. Joshi had a distinguished academic career at the Department of Genetics and Plant Breeding, Institute of Agricultural Sciences, Banaras Hindu University, where he served as lecturer, senior scale lecturer, reader, and professor between 1985 and 2016. Through his teaching, research, and mentorship, he contributed significantly to capacity building in plant breeding and agricultural sciences. Dr. Joshi has received several prestigious honors, including the Jeannie Borlaug Lobe Woman in Triticum WIT Mentor Award in 2014 from the Borlaug Global Rust Initiative at Cornell University, recognizing his exceptional mentorship and leadership in wheat research. He also received the V.S. Mathur Memorial Award from ICR, Indian Institute of Wheat and Barley Research, for his noticeable contributions to wheat improvement. He is an elected fellow of several premier scientific academies and societies, including Indian National Science Academy, the National Academy of Sciences India, the Indian Society of Genetics and Plant Breeding, and the Society for Advancement of Wheat Research. Dr. Joshi's research has had a major impact on global wheat improvements programs, particularly in the area of disease resistance, climate resilience, and seed delivery systems. His specific leadership and commitment to sustainable agriculture continue to support food and national security across developing regions of the world. With these words, I once again extend a warm welcome to Dr. Arun Kumar Joshi, sir, and invite him to addressing the gathering. Over to you, sir.
Thank you so much, sir, for joining us in your busy schedule. Please.
Karthik, sir, unmute yourself.
Yeah.
Thank you. No, I think he's correct, So I'm here because of Karthik, he contacted me, so because he was working with us. Thank you very much, Mayal. Thank you very much. So, dear friends, esteemed colleagues, see, I cannot see you from this window, but I know you are already very experienced and well-known scientists in your field. I will share the PowerPoint. And I believe you can see this. Karthik, you have to confirm. You can see.
Yes,
Ah,
sir.
yes, sir. Yes, we can see, sir.
Slide is
Yes.
visible, sir.
Slide is
Okay.
visible. Thank you, sir.
And
You can
you
proceed.
can hear also.
Yeah, sure. Yes, sir. You are audible.
Thank you.
Thank you.
Thank you. So, dear friends, I have 30 minutes. I will tell you a couple of things which maybe that you know very well. Well, there's nothing very new about it. But the topic I got from your organization. I'm really grateful to your organization. MET is very prestigious organization.
Sir, please unmute yourself.
Again,
Sir, please unmute here.
somebody is muting me there. Can you hear me?
Ah, yes, yes.
I think
Now we can hear. Yeah.
it from that side, Anyway, so I got an email from your prestigious organization. Can I speak on something on climate resilient technologies? See, you heard about my background, but in last 10 years, I was wheat breeder, wheat geneticist. I was working in seed science. But then last 10 years, I worked on climate resilient technologies when I was in CIMMYT and Borlaug Institute for South Asia. So I have taken a simple topic, how to empower farmers through climate resilient technologies for sustainable growth. I will give you some examples, not too many. And I believe that my talk will be able to give you some, something new, some thoughts for future. So here, the two keywords you can see: farmer and climate resilience. Now, why about farmer? Because farmer is worried. So we are also worried. It's not that only farmer is worried. So why farmer is worried? Farmer is worried. When I was searching, I was interviewing farmers, I was searching websites. I found answer in FAO website very recently. This website says that for an individual farmer, the biggest fear is climate change, extreme weather and climate. Of course, he's worried for input costs, supply chain, profitability, debt, and there's so many. We are also worried for so many reasons. But farmer is worried because he cannot predict what type of climate. And climate change is happening. What are the problems. Temperature is increasing. Our climate, Earth, about 100 years ago, it was 1.5 degrees centigrade cooler. We are moving towards warming. And recently there was a survey was made. Survey was made by UNDP, and it was found that about 50% people are more worried about climate change than last year. This is called People's Climate Vote. It was conducted in 77 countries and 73,000 people participated. So this is biggest climate vote done so far. It was carried out by UNDP and University of Oxford. So not only farmers are worried, everyone is worried, and there are genuine reasons for that. I'm not telling you the details of those things. It means we need in agriculture something on climate resilience. So we need climate resilient technologies. That's very simple definition. You can define in different ways. You can say it's climate smart agriculture. You can say climate resilient agriculture. There are different definitions of these keywords. But actually they are practices and techniques that enable farmers to adapt to and mitigate the impacts of climate change. Generally, it is an integrated approach. Use whatever is more useful, whatever is available, whatever you can procure, purchase, understand, and use. You cannot define it. What is this? You can name 20 things. It can be only one. But here, sustainability is very important. Now, for example, in summers, umbrella is enough. for sun, not for rain. Rain is different. Umbrella for rain used to be used, a lot. But now we use umbrella for even for sun. It's very common now. So anything which we can use, so you can list large number of these technologies. It can be a seed which is stress tolerant. It can be resource conservation technology, crop animal model, biodiversity, precision machines. Now AI is coming, finance, insurance, advisory, capacity building. It can be anything. It will vary from country to country, region to region, village to village, person to person. So this photo is from Bisa. He is using a mobile tool to record something. Anyway, so from where we are getting this information? I am not traveling each and every country. You are not traveling. From publications. So I was searching estimated publications on climate resilience. I found that from 16 to 26, see the growth, 1,000 to 12,000. So every year, a lot of publications are coming. They're growing at the speed of about 25% per year, even more sometime. So these publications are talking about nothing but how to fight climate change, how to help farmers, how to help the entire industry, the entire country, the GDP. And then I was searching what are the keywords which are used here. I found that mainly it was energy. In climate change is coming because of energy. So word cloud, let me use this. Let me use, where is this? How to use a marker more. Because if I have a layout, slide control, I think I'm not getting marker here. Anyway, so you can see word cloud illustrating key sustainability, energy, efficiency concepts where word size represents relative frequency. So in this literature, search of literature, which is not moving next. Okay. So actually there were 51 research articles from 2009 to 25. They were examined to determine technology trends, performance, outcomes, and challenges by Gasa et al. And he found that it actually, whatever we are doing is because of, their energy is very important. But you can see sustainable agriculture, environmental economics, cultivation, sustainability, alternative agriculture, greenhouse gas. So many words are here, but somehow I found one word, China, one country here. For some reason, maybe they are publishing more, that's why. So you can see in the left-hand corner, I'm unable to use that pointer actually here. So I'm trying to search where is that pointer. So if I get that pointer, it will help me. More record transcript.
Maybe you can use cursor, sir.
My cursor you can see?
I think yes, you can.
You can see.
Just indicate
Okay.
by cursor.
Okay. So based on these publications, and then it was concluded by this author that there are two significant innovation pathways. One is renewable energy technology. It can be anything. It can be solar, it can be anything. Next is digital solutions such as precision agriculture, Internet of Things, enabled monitoring, and automation. So this is something new. 20 years ago, these things were not so frequently cited. But anyway, so how do innovations help? You know how innovations, I will give you some examples. And you must have seen that example from University of California. They said that their publication says that their artificial intelligence weather prediction helped Indian farmers last year. India's Ministry of Agriculture sent regular monsoon forecasts by phone to 38 million farmers in northeastern India 2025. Because in northeastern India, irrigation facilities are at very low level. Punjab, Haryana, much better. But in eastern India, eastern UP to Bihar, other places, irrigation facilities are at very low level. And for the first time, farmers got up to 4 weeks advance notice before the continuous rains, helping them time crop planting better. So it helped. Actually, it helped last year, and very accurately. Actually, the point here is accuracy. Each year you must be seeing weather forecast, weather predictions. But last year it was very, very accurate. They made a model, sent it to Government of India. They circulated, gentlemen, the rain will come like this. And exactly same thing happened. So farmer got 1 month to plan whether they should go for rice or any other crop, when to plant nursery, when to do other operations. So this was very useful and very economical in general. If Government of India or any government spends $1, you will get $100. So this is one technology, how climate resilience can be achieved. Now this is another. Now you must have seen this. China is using bacteria. This is a very new, a new use. This came in Economic Times, May 26. The news is very new. China is using a bacteria to turn desert into fertile soil in just 10 months. Now, validation has to be done here, but they call it a breakthrough. Researchers at Shabtao Desert Experimental Research Station, they use cyanobacteria. They are saying that they can create biological soil crust, a living layer that stabilizes shifting desert sand and helps vegetation grow in extremely dry conditions. Now, some colleagues may say, is this agriculture? No, this is agriculture. Even grass is agriculture. See the business of the grass in the world. If you include turf grass, it is $100 billion. India has 5 million hectares of more than that desert. If we can reclaim, we can get that soil back. Actually, it is a very big area. Now, another important thing is FAO. It's very new. FAO advances new technology to monitor soil fertility. This is mid-infrared spectroscopy. Now, this is very, very efficient technology. There are several soil testing mechanisms. Farmer doesn't test any soil. His way of doing is different. He observes number of birds in the field while plowing. If more birds are there, it means my soil is very fertile, because there are earthworms and different kinds of living organisms after which birds do come. But you don't know, you can, by this technology, you can very accurately predict organic carbon, texture, nutrients. And this was done by soil firm in soil mapping for resilient agri-food system. Now, why I'm giving you this example? Because this is very, very efficient system. There are several machines, tools, lab methods are available. They are not so efficient. They take a lot of time. This method, within few minutes, can give you sample of hundreds, the result of hundreds of samples. Now, these things are very important. Without monitoring soil fertility, you cannot move. I'm giving you one example. This will help in many ways because, because of wrong crop rotations, our fertility is going down. So we cannot do anything. So after that, you can start doing something. Now, systematic approach to mechanization. Why I'm showing you this? Because mechanization is very important and rural landscapes are not same. They vary from country to country, state to state, region to region, village to village. You need bigger machine, lesser leveling. You need smaller machines. You need, post-harvesting, you need before harvesting, and you, on the right-hand top, you can see, what is this? A mechanic in Kenya. So systematic approach to mechanization means you need mechanic. Don't think you need only machine. Without mechanic, you cannot do anything. A driver means driver come mechanic. If there is no mechanic, no mechanization will move. So this mechanization is very, very important. It varies from place to place. And I will show you a simple example you must have seen somewhere. This is from CSA project. And this result is based on about 4,000 sites in Eastern India. It says that early sowing of wheat gives enormous advantage. So how you will obtain it? You will achieve it by combination of variety and a machine, zero-till machine. So if you plant in November, you get higher yield. If you plant in December, you get lower yield. So if you have a machine, you can plant early along with the right seed. This is normal maturing wheat variety initially, then early maturing wheat variety. So these are proven studies which say that you don't need a rocket science. You need a simple machine, a good seed, and an advisory. And you can see 2.68, here is 5.36. This is average. Some farmers got 7 tons. So this is how you can adjust your planting, and you can then reduce the effect of climate change, the global warming. Now, another story is early heat tolerance in wheat is a new success story in northwestern India. Earlier, I told about eastern India. Now, this is about genetics. October sowing proved that with a simple genetic change, there is a gain of 1 ton per hectare in wheat with same water, same nutrition. So there is genetics involved in that. Earlier, there was no early heat tolerance. But you can see this paper published in 2021, along with ICR, BISA, CIMIT combination, we produced a result which says that early heat tolerant varieties give higher yields. 540 lines when tested in 2 conditions, they proved, yes, there is genetic heat tolerance during early growth. And 3 varieties were released in India in 2020, about a dozen since then. So simple changes can help us fight climate change. Now, I am showing you this field, new mode of action, herbicide. Now, what is this? This is weedicide free. If you don't put weedicide, you can see, what is this? This is wheat field. These are nothing but only weeds. And when you treat weedicide, you can see your crop. So weed is a very, very serious problem. So weeds are a serious issue in crop production. We need a new mode of action in herbicide, as herbicide resistance is a major issue. Some people will say that herbicide is not sustainable. Then you can use integrated approach. And now you can use a non-GM herbicide tolerant crop approach. The examples in rice, you must have heard. In wheat, they use an altered ALS gene. So because of that, they use the weedicide, imase, and crop loss is not there. It kills only weed, not your crop. You must have read. So there are, because weeds are, what I want to tell you, weeds are very, very important. And in climate change, weeds are adjusting very fast. We are not doing any breeding for weeds. They are doing breeding naturally. So they are growing very fast. So we have to do something. Now, what is the evidence that there is a progress? There is climate change. Last 20 years, what is the evidence? The evidence is this. World agriculture output, price, and you can see here, I'm trying to tell this is the agricultural price index, 1900 to 2000. Price is not going down actually. Population is going up, production is going up, and the land is same, almost same. So the message is food production is higher than population growth, and food is available in affordable price. Despite so many problems, it is going up and up. It means there is something in the technology, there is something in our policy, there is something in our entire approach. So cropland has increased only marginally, but food is available in affordable price. Of course, recently, last 2-3 months, we are seeing some problem of energy and they are fighting anyway. These things do happen. Now, value of world agriculture output shifted from global north to global south. What I want to tell you, you know global north and global south? Global south means Africa, Asia, poor countries. Latin America, poor countries. Global north, rich countries. The global south was considered traditional but grew faster due to the integration of modern approaches. So what is interesting, that global south, you can see the value of agriculture product is more here. Of course, population is increasing global south, but you can see in the total world production, agri production, whatever, you can see that global south is now prosperous in terms of food production. Availability can be a different issue. We will discuss that. Now, diffusion of varieties. Diffusion of varieties, you can see here, 60%. In the entire world, this global South Asia, this report I have taken from USD Economic Research Service, you can see in 60% area is covered by modern varieties in the global South. So these varieties are helping. They are drought tolerant, they are heat tolerant, they have good quality, they have, even they are fertilizer efficient, not to that extent, but they are machine, they can adjust to machine, they can adjust to intercropping, they can adjust to mixed cropping, whatever, they can adjust to different dates of sowings. There are a lot of things here. And both traditional and modern agriculture emphasize the need for good seed. Good seed is something historic, but resilient seed is the basis of resilient agriculture. Without this, nothing is possible. Another important thing is biofortified crops. Biofortified crops means nutritional crops, you know all that. So in last about 15 years, 491 biofortified varieties have been released in Asia and Africa. Different crops, sorghum, zinc sorghum, sweet potato, rice, pearl millet, wheat, maize, lentil. This is also important because nutrition is a very big problem. And in climate change, when the temperature will go up, when there will be problems, we need more nutrition. Not only we, animal husbandry. Our cattle, they need better, nutrition. So when a crop is biofortified, certainly it is biofortified. The straw is also biofortified. It has more nutrition. It has been already observed. Now, this is very interesting. Farm labor is decreasing. You can see around 2010, you can see the world agriculture labor is left side, world agriculture capital is stock, right hand side. You can see this stock is increasing. Agriculture capital stock. What is agriculture capital stock? In your farm, the entire machinery, your processing center. This is your capital stock. But labor is going down. Why? Because of machinery. So mechanization has increased in countries where agriculture was considered not so mechanized. So because of this reason now, the farm labor is decreasing. While agriculture capital investments are increasing, it means machinery use is increasing. Now, another thing is, here is the message. Improved practices are saving water and reducing emissions. Now, these are the graph, 1960 to 2000. World cropland and water use intensity, it is going down. And you can see the tons of carbon dioxide equivalent to $1,000 of agriculture output is also going down. So emissions are going down. There is data on this, but this is not enough. We are saving water, we are reducing emissions, but population is growing, is still growing, food demand is growing. So we have to do much more than this. This is the message. So why I'm saying that? Because there are challenges. We are progressing very nice, everything good, looks fine, okay, but population increasing. What is the problem? The challenge is, this is a very big challenge for a country like India. Our farmers are small and marginal. There are too many small and marginal farmers. 90% are in the category of small and marginal. Small and marginal means less than 1 hectare. Less than 1 hectare means less income. Average income of a farming household in India is how much? According to a report from ICRIER, January 2026, government data is same. If you see the press release by Press Information Bureau, which records discussion in the parliament, almost same. They are telling data about 5 years older, 2019-20 something. So average income is how much? 13,000 to 19,000. Only crop, only 4,500. If you are taking only crop, farmer cannot survive. He is below poverty line. The alternative sources he has to use. He depends on animal husbandry 12%, wage labor 16%, government job, enterprises, doing something, a gumti, a small shop, 15%, private sector work, some job, some teaching, some here, some consultancy, some help. Otherwise, farmer is really poor. You know where the farmers are happiest in the world? I think you go search Google, you will find different answers. But when I travel to different countries, I was asking, are you happy? Mexico? Oh, okay, happy. Kenya? Oh, not so happy. Ethiopia? No. All Zambia, all those countries, Argentina, Brazil, South Asia, India, Pakistan, Bangladesh, Bhutan, Turkey, traveled whole world. Japan. Then I went Japan. Of course, Europe also. Oh, nothing to ask in Europe. Still, the urban and rural divide in some countries. Urban people say, oh, they are enjoying subsidy, and villagers say, oh, I'm suffering. This is problem US also. So yeah, I went Japan. I found them happy. You know why? Because there is no farmer in Japan, according to information I got, who is not doing a job or a business along with agriculture. Same thing in India. Only crop cannot feed you. So these are the challenges. So what is the problem? Then farmer cannot take risk. You cannot go and tell him, use this drone, use this machine, use this aircraft. Oh my God, I will give you this mobile, that. No, sir, sorry. You need bottom-up approach. Ask them, discuss with them. How can I help you? Tell me one thing. He will say seed. Tell me second thing, improve my machine. Tell me third thing, he will say, this is my irrigation system. Without cost, can you improve? Only then farmer will be happy. Another problem, meat consumption is growing on. It has significantly increased the demand of 70% more food production by 2050. Because producing meat is highly resource intensive, It requires at least 10 kg of grain to produce 1 kg of meat. Actually more, 15, 20, 25, 30 kg for 1 kg of meat. Cheapest is chicken. This puts immense strain on land and water resources. Why this figure is lower side? In villages, the poultry, it's open poultry. They don't give too much of grain. They just move like, cows and buffaloes, and they come back. Industry has to feed them. And you can see these countries. Where is India in this? In the bottom. India is here. Pakistan, third number. China in the middle. On top, Australia. Where is USA? USA is not here. So you can see some countries, the meat consumption is increasing. So first solution for the problem of the world is please become vegetarian. If you are vegetarian, congratulations. Please become vegetarian. This is the answer. Now, thought for you. I want to give you some thought so that you can, how much time I have? How much time I have? No, 2 minutes. Farmers were always smart. They developed resilient technology since day one. Example, land races of the crop. Land races are synonymous with stress tolerance. So please do not think farmers were not smart earlier. They were always smart. Always. What is this? Indus Valley civilization. It was marvel of ancient agronomy. They had sophisticated water management, strategic crop planning, crop rotation, storage system. They were growing, earliest evidence of domestication of cotton. The farmers were smart. They are smart. They will continue to be smart. So let us not underestimate them. How many companies are there in climate tech? I was searching globally about more than 7,000. In last year, 1,600 created. Value of 1 to 10 billion, 15. India in climate tech ranks third, about 262 companies, mostly on seed, 20% very risky venture series. This is early stage ecosystem. They are dominant in Maharashtra, Karnataka, and Delhi. What is this? Why I'm showing you International Year of Rangeland and Pastoralists. Do you know this? Which year is International Year for them? 2026, this year by United Nations. But why? Why rangeland? I am not showing you cropland because they cover half of the world. They are home to unique biodiversity. They hold diverse cultural heritage, indigenous knowledge, and preserve ecosystem across the world. All Hollywood, Bollywood music is coming from there. I don't know whether they get royalty. I don't know. It's like reserve bank of our heritage. And in India, climate resilience in Indian rangeland, the strategy includes three things: rotational grazing to prevent overgrazing, cultivating drought-tolerant fodder, and integrating livestock with agroforestry. My message is, don't think only crops are agriculture. Rangeland is also agriculture. It's a very big area. So what is the way forward? I think time is going up. Way forward and conclusion. The way forward and conclusion is that one technology will not help. This is example from climate smart agriculture in maize in Tanzania, that a number of practices help maize, whether water harvesting, rice management, reduced tillage, organic matter, intercropping, inorganic fertilizer, improved variety, green manure, several things. So everything combines, then only you can get a potential of 100% of a crop. So one technology will not help. You cannot see from your eyes the benefit. You have to add one, then two, then three, like a layer of onion, then it becomes onion. So in my last slide is Global Environment Outlook, highlights 5 solutions for the world in fighting or in having climate resilience. Number 1, cut down meat and boosting plant means become vegetarian. Use plant-based foods. Farming and fishing to become more efficient. Government, business, etc. You cut food waste. Food waste is up to 30% in some places. And try new food, lab-grown meat. Have you seen artificial meat? It's very common now. Artificial meat, artificial salmon, artificial fish, and vertical farming. It's much better. And food systems should focus on local produce. Don't bring your fruit from Chile or Switzerland. Consume locally. Diversify supply chain locally. We are bringing, kiwi. Kiwi is coming from where? Switzerland and Chile. Oh my God, Latin America. And they are rotting in Arunachal Pradesh because we don't have packaging system. Anyway, so these were the solutions we are given by United Nations Agency very recently in December 25. And with this, I think I must close, and I thank you all for your patient hearing. Thank you.
Okay, sir. Thank you.
Thank you very much, sir, for your enlightening and highly informative lecture. We sincerely appreciate the valuable knowledge and insights you have shared with us today. We shall now proceed towards the question answer session. Participants are requested to post their questions in the chat box.
Audience, do you have any questions? You may ask if you have any questions related to heat stress, biotics tolerance, abiotics tolerance. Professor A.K. Joshi is familiar with this kind of study.
Yeah, you can ask. I think something is in chat box. Something is there.
Oh.
No.
Sir, I have very good afternoon, sir.
Yes, please.
This
Namaste.
is
Yes, please.
Dr. Abhay Sharma, working as a research associate at Horticulture Research Station, Kullu, Himachal Pradesh.
Ah,
First of all, sir, congratulations on delivering such an insightful and thought-provoking presentation, sir. I would like to share with all the participants that I'm associated with a project, Gramin Krishi Mausam Seva, sir, which is funded by IMD New Delhi. And what we do is we disseminate location-specific 5-day weather forecast and agro advisories to the farmer community. As you have also discussed with that weather forecast in the previous year, it was very good. That's what we are doing, sir. We are enabling them to make timely and informed decisions regarding sowing, irrigation, and fertilizer application, and all the stages are covered from sowing to harvest. And the other thing that I want to share that these advisories actually, with the help of weather forecast and are adaptive to climate change, improve resilience of agriculture system, and support the broader goal of achieving sustainable and climate resilient agriculture for present and future generations. That's what I wanted to share, that how agrometeorology serves a bridge between weather science and farming, helping farmer community to achieve sustainability. So thank you, sir.
Yeah, thank you. So your name is Abhay.
Yes, sir.
Abhay ji, thank you very much. I am very glad that you are involved in that. See, farmer always will be helped if you give him advance information, but also give them some solution.
Yes, sir. Sure, sir.
Thank you.
Professor Enrico Porcedo want to ask
Yeah,
from
yeah.
Italy. Sir, could you please ask your question? Professor Enrico Porcedo.
Yes, thanks. Very much in line with my thoughts are very much in line with your ideas you presented. My point to university staff is this. The system needs to be governed. Is the government ready to do this job? As the pieces of information available, as university provided or is going to provide information on how to set policies. Thank you.
Thank you. Well, I think your question has already built-in answer in that. Government is helping, it will help, but we all have to work together. Scientists have to come forward with innovations. For example, there is a wonderful question here. I saw one question, sir. What you see as the most promising tool of biotechnology which can help agriculture in next decade? Anybody can answer that. It is nothing but gene editing. Use gene editing. You can use gene editing anywhere now. The way it is growing, it is like AI.
So whatever, sir, but for durable resistant, it is still impossible. In the case of biotic stress, it will, because pathogens will arise again and again. So do you comment anything on that?
See, it's very simple, the warfare you are seeing, the world is fighting, this is also biological, there is host and pathogen. They evolve, keep on evolving. So you have to be very intelligent. Pathogen is more intelligent than us because it is living in nature. So if you are using one gene, it will get defeated. 2 gene, chance will be 50%. Yes. 3 gene, it will. So it is like multi-gene. When you are
pyramiding,
pyramiding, so there are cassettes now. So you can edit multiple genes, combine into one cassette, insert somewhere. Science is growing. Pathogen is very intelligent. There can be super race. Remember, if you cannot eradicate a pathogen, you will try to eradicate, it will become even bigger, more powerful. So you have to make a balance, make a balance. Yeah. Keep pathogen also happy. Yes.
Any questions from audience? Professor DK, sir, do you have any questions?
Yeah, I'm thinking, Arun sir, Nice to see you over here on this platform. Sir, I was thinking that the peoples are deploying various approaches like genetics and plant breeding, genetic engineering, and alternatively, if we talk about the plant microbe interactions, that may help crop to grow under abiotic and abiotic stresses like pathogen-free crop must be there, and the temperature tolerant, psychrotolerant genes, heat shock proteins must be expressed in such crops. So what do you think that which one is the best approach whereby the farmers may deploy henceforth?
No, see, farmers, farmer is a very simple man. He will come to you, sir, give me a magic seed.
Yeah.
Which will require less water, which will be heat tolerant, which will give me more yield, better quality, better market. So farmer will come with a simple question. Breeder has to now plan what to do. Now breeder, sometimes there are different types of breeders, and there is always lab research, field research. What we have done is field research. We do nothing. We simply grow large number of lines in the field under that stress, find out the best one, start crossing, and we produce the highest yielding under that condition, less irrigation, or we don't measure heat shock protein. Or this is for MSc PhD thesis and a research which will help us 20 years later, because
Right.
this pure science is very, very important. Our students, our colleagues should understand whatever Einstein did is helping today. So basic research is very, very, I do agree with you. So we have to do both applied research, basic research, long term, medium term, short term.
Right.
This is
Basic
good question.
and
Thank
applied.
you. Pleasure.
Okay, thank you, sir. Thank you.
Thank you.
Thank you, sir. Very nice question.
I think there are lots of any question in the chat box. No, after biotechnology, no question. Anyway, okay. Thank you.
Thank you, sir, for patiently addressing all the queries and for sharing your valuable perspective with our participants. Your responses have greatly enriched today's session and provided deeper insights into the subject. With this, we come to an end of the question answer session. On the behalf of the organizing committee and all the participants, I extend our heartfelt gratitude to Dr. Arun Kumar Joshi, sir, for his inspiring lecture and generous interaction with us. Once again, thank you so much, sir.
Thank you.
Thank you,
Chief.
Professor. Thank you so much for joining. Thank you
Thank
so
you,
much.
everyone.
Thank you.
Bye-bye. See you.
Thank you, sir. See you.
As we continue with the session, it is my immense pleasure to introduce our next prominent speaker, Dr. Bhupender Giri.
Dr. Payal, your voice is very low. You can check your computer or somewhere.
Dr. Payal, remove headphones and
I think
use
your voice is very
computer
lower.
full voice speaker.
Okay, sir.
We are not able to hear you, Payal.
Yeah, you can just talk. We will see.
Sir, am I audible now?
Now,
It
yes.
is fine.
I thought it will change, but yeah.
It is
Okay.
fine.
It is.
As we continue with the session, it is my immense pleasure to introduce our next prominent speaker, Dr. Bhupender Giri. He is a distinguished academician and researcher in the field of plant microbe interactions, microbial biology, soil microbiology, and sustainable agriculture. He is currently serving as professor in the Department of Botany, University of Delhi. With over 2 decades of teaching and research experience, he has made significant contributions in understanding the role of mycorrhizal fungi in salinity tolerance, drought resistance, soil health, and climate resilient agriculture. Dr. Giri completed his PhD from the Department of Botany, University of Delhi in 2001 and pursued postdoctoral research at the Department of Plant Pathology, North Carolina State University, USA. His research has received wide international recognition with over 5,700 citations, HI index of 25, and more than 48 research publications in reputed international journals. He has edited several internationally acclaimed books published by Springer Nature, including Root Biology, Biofertilizers for Sustainable Agriculture and Environment, Microorganism in Saline Environment, and Soil Health. His research focuses on microbial biotechnology, plant stress biology, soil carbon sequestration, and sustainable agricultural systems. Dr. Giri has successfully completed several funded research projects supported by agencies including DBT and UGC, particularly on mycorrhizal technology, saline soil reclamation, and sustainable agriculture. He has delivered invited lectures and chaired scientific sessions in prestigious international conferences across Denmark, USA, UK, Switzerland, and Canada. He is a recipient of Raman Postdoc Fellowship, DST Young Scientist Fast Track Project, and the Best Poster Presentation Award at the Indian Science Congress. One of his landmark review papers published in Annals of Botany was recognized among the top 10 cited publications in biological sciences and agriculture in India. Dr. Giri actively served as reviewer and guest editor for several leading international journals and is associated with numerous professional scientific societies. His work continues to contribute significantly towards climate smart agriculture, soil sustainability, and microbial biotechnology for environmental resilience. I now invite Dr. Bhupender Giri sir to deliver his lecture and share his valuable insights with us. Over to you, sir.
Thank you. Thank you very much for very nice introduction and very good afternoon, everybody. My dear friend D.K. Chaudhary, Dr. Payal, Dr. Karthikeyan, and all the faculty members who are available at this time. Very good afternoon, all of you.
Good afternoon, sir. Please.
First of all, let me know that how I can
Share. You can share at your end.
Okay.
Share option is there on below.
Share, yeah. This is here. And so I'll go to the document.
Certainly.
Okay.
From where you have saved your file, you can click over there. It will show the reflection over here.
Okay. My files.
Yes, it is loading now.
Let it take a few moments.
Certainly. No, it does not appear, sir. If you are in system, you can mail us.
Okay, just a minute. Let me.
No. No, it does not work. Again, go back, share, open your folder and click on the save file.
There is also a share option here.
Do you see share option, sir?
Yeah. There is a share.
The taskbar is there at the black color.
In your computer, you can just open, sir. It will appear directly. Just
I opened it.
Then you can go to share. You can see below share option is there in the Zoom.
Yeah. There is the share option.
Just click that, sir.
Okay. Why it is not coming? In case of Google Meet, it's very simple.
No, sir, if you click share, you can see various files directly. It will appear. Then you can select.
Okay, I go to the file. मैं मेल करता हूँ फिर आपको, ये नहीं।
yeah, you can please send us
You do
email.
mail on revert to
हाँ।
Karthik.
Yes, yes.
Please, we are
हाँ।
waiting for your mail.
Yeah,
कौन
just
से मेल पर करूँ, डीके आपके पर कर दूं?
Sure,
yes, sir.
you
Send
can
you.
do it.
आई एम सेंडिंग इट टू डीके चौधरी.
Okay, sir.
Okay.
Sir, please check.
हैव यू गॉट इट? नहीं नहीं, अभी जा रहा है.
Sir, if you want to share, just open your file at your
Uh-huh.
desktop.
I
And
open my file, there is a
Yeah.
share
Just-
option, but when I go to the share option,
yes.
what it shows name of the file, place, but it is not showing uploading option.
No,
That's
no,
why it's not.
you have to open your PowerPoint, sir. Then only it will show.
Okay.
You have to
PowerPoint
open.
में PowerPoint
Yeah,
type
it should be opened. Then only it will show.
सर PowerPoint में ही मैंने किया है, उसी का ही
Open,
बता
if you open, it will show, sir. You can open the file.
File is quite longer, that's why it's not reaching you quickly. Oh.
Sir, just open your file, PPT, then click share option, sir.
सर मैंने वो कर लिया, नहीं हो रहा है उसका, वो दे नहीं रहा है, फिर ऑप्शंस नहीं दे रहा है उसके अटैच करने का, मतलब उसको ओपन करने का, ना अपलोड करने का ऑप्शन दे रहा है.
Your mail did not arrive to us, sir. We are waiting for your mail.
हाँ मैंने भेज दिया. एक्चुअली फाइल इज क्वाइट लार्जर, दैट्स व्हाई इट इज टेकिंग टाइम.
That's why it takes time.
Yeah.
It was very easy on Zoom to upload the system, right? It was very easy.
मैंने दोबारा भी भेजा यार, हो नहीं रहा है.
Sir, आराम
See,
से.
I opened my
आपने
PowerPoint
डॉक्यूमेंट ओपन कर लिया? आप इट्स
Yes.
PDF or it's
There is the share option.
हाँ, just
Move
click
or
on share.
copy this presentation to the cloud to share. This is showing.
सर इसमें एंटायर स्क्रीन आ रहा होगा और एप्लीकेशन विंडो आ रहा होगा.
नहीं
ऐसे
आ रहा.
स्क्रीन खुली होगी ना, एक स्क्रीन खुली होगी, सिलेक्ट अ विंडो और एन एप्लीकेशन दैट यू वांट टू शेयर, ऐसे करके लिखा आ रहा होगा सबसे ऊपर.
नहीं, actually it's a Mac. It's not.
Oh, okay, it's a Mac. Oh,
Yeah.
okay.
It's a Mac. पहुंचा आपके पास, पहुंचा नहीं?
Not yet, sir. We are waiting.
Oh. ठीक है, चला गया है, चेक. मेरे यहाँ सेंड फाइल में दिखा रहा है. Yes.
Send
Now
us
check.
PDF
Check.
file, not PPT.
I'm checking, sir. I'm checking, but not arrived at my end.
यहाँ से क्लियर हो गया है, सेंट मेल में आ रहा है मेरे में.
Which mail ID did you use? DK
I show you.
Chaudhary 1?
Yeah, I let you know.
Because there is another DK Chaudhary in Amity.
Devinder Kumar Chaudhary, DK Chaudhary 1 at the
Okay.
MIT. I
Yeah.
know you only.
Yes. Reciprocal, sir. Reciprocal. Anyways, we are waiting for you.
Maybe you can
नहीं, यहाँ
send
से
us
मेरा सेंट
PDF
मेल में
file,
क्लियर
sir.
आ रहा है, मिल जाएगा भी आपको.
Otherwise, you can send us PDF file. It would be easy for us.
You can just
PDF. Okay.
Yeah.
Okay.
You let
It
it
will
try.
be easy.
Yes,
Okay,
we received your mail, sir. We received your mail.
We will check. We are
Okay.
checking.
We are going to check whether it opens or
So
not.
there is no need of PDF now.
No need.
No, we will check, sir.
Let us check.
Yeah, click to download.
Click to download.
One more time. No, not this one. No.
File is lengthy, 20 MB file it is. We can share it. File has opened, sir.
Okay, sir, we are sharing. 1 minute.
We are
Okay,
trying to
okay.
share
Good,
with
good,
you.
good, good. At last.
This one, yeah. This one.
Okay, sir.
It opens now.
Now, yeah.
Yes.
It's open.
reflects.
It's open. Thank you. Thank you.
Yeah. For each slide, you can just say next to us so we can
We will
operate.
move ahead. Okay.
Okay. Thank you. Thank you. So again, very good afternoon for a
So, sir, unmute.
Sir, please unmute yourself.
Okay.
Are you able to hear me now? Am I audible now?
Yes, sir, you are audible.
Okay. Thank you. Thank you very much. So, dear friends, my lecture is on shielding plants from environmental stress, the power of microbial inoculant. What I'm going to tell you in this lecture is that how the microbes can help us in this crisis of climate change and the soil which is facing the plants which are facing the problem of abiotic stresses particularly or environmental stresses. So for last about 25 or 28 years, I'm working on a fungus that is mycorrhizal fungi. Beside this, I'm also working some other kind of the microbes which are helpful for the plants to improve the growth of the plant under stress conditions. That's why I'm taking you to this journey, and I'll let you know that what kind of the benefits we can get from the microbes which are present in the soil. Sir, how I will move? Okay, thank you. So, what is the mycorrhiza? Mycorrhiza is a mutually beneficial symbiotic association between soil, fungi, and land plants, where fungus assist host plant to obtain nutrients from soil, and in exchange, the fungus gets food that we call the carbohydrate from the plant. So I think several of my colleagues will be knowing about this fungus, that what the fungus is doing in the soil. So they are helping each other, means host and fungus, they are getting benefit from each other. सर मुझे बताइए इसको मैं आगे कहां से बढ़ाऊंगा, इसमें कोई ऑप्शन नहीं आ रहा मुझे आगे बढ़ाने का. So there are different types of mycorrhizal fungi which are occurring and making association with the plant root. This slide shows you that about 22% of vascular plants are vascular mycorrhizal and 1.5% are ericoid mycorrhizal, whereas 10% are orchid mycorrhizal plants. And only 2% are ectomycorrhizal plants. Just 8% of plants are completely non-mycorrhizal. In fact, there are some plants which are even the seeds of those plants are even not able to germinate without this association. So they are totally dependent for their germination. And 7% have inconsistent non-mycorrhizal or vascular mycorrhizal association. So in this slide or in this picture, you can see that this is the ectomycorrhiza, which is colonizing the plant surface. This is the avascular mycorrhiza, which is not only at the plant surface or epidermal cells. In fact, it is entering into the cortical region of the plant root, where it is making certain specific kind of the coils. And there are the structures which we better know as profusely branched structures, which are the arbuscules. Please, abhi back slide par jaiye, sir. Go to the back slide. Yeah. And another is monotropoid, which is making peg-like structures in the epidermal cell of the plant root. Arbutoid mycorrhiza, which is also making coil, but these coils are limited up to the epidermal cells of the plant, not in the cortical region. While ericoid mycorrhiza and orchidoid mycorrhiza are going little inside of the plant root. Next, please. If I see the classification of mycorrhiza, it is very interesting that at different times, different people have classified mycorrhiza into different groups or orders, or in fact, I can say the phylum also. If you see, in 1974, Jerdman and Trappi, they classified mycorrhiza into phylum Zygomycota with one order, one family, and 7 genera only. Later, in 1990, Morton and Benny, they also classified, they gave the another classification where the phylum was remains same, it was Zygomycota, but they increased the number of orders. So 2 orders and 3 families with 6 genera, they classified it. In 2004, Shovler classified them. Shovler raised and his colleagues raised the order Glomales to the rank of phylum Glomeromycota, where 4 orders, 7 families, and 7 genera they decided to have. And the last one was given in 2011 and 2012 by Ohl and Goto, where they added the order Gigasporales to phylum Glomeromycota. And there were total 5 orders, 15 families, and 30 genera. So in this way, the latest classification is having order Gigasporales also in the phylum Glomeromycota. Next, please. How we can identify the mycorrhiza inside the plant root? So there are 2 very peculiar structures. On the basis of that, we can say that this is the arbuscular mycorrhizal fungi. Particularly, I'm talking about I am working on the arbuscular mycorrhizal fungi. I have shown you that there are different types of mycorrhiza which are making association with the different families of the plant. But in this lecture, I am focused on the arbuscular mycorrhizal fungi only. So arbuscular mycorrhizal fungi derives its name from Latin word arbusculum, which means a little tree. They are obligate symbionts with no confirmed sexual stage. So remember, this fungus is not having a confirmed sexual stage. They have 2 types of mycelium. One is multifunctional extraradical mycelium and another is interradical mycelium. And the hyphae produce dichotomously branched arbuscules. This is the structure of arbuscule, which is formed in the cortical cell of the plant. Next, please. Another structure is the arbuscules, which are pear-shaped structures, which are known as vesicles. They are present in the cortical region of the plant root, and they are looking like the balloons. So AMF hyphae produce balloon-like structures known as vesicles. And in this slide, you can see there is the extending hyphae, and these are the arbuscules in the plant root. Next, please. These are the fungal spores, AM fungal spores, which are present in the soil, not in the plant root. They are identified on the basis of their structures, morphological structures, their subtending hyphae, how the hyphae is arising, and what kind of the shape it is taking, and what is the shape of the cell, the walls of the spores. So different morphological structures are there. On the basis of those, they are identified. But beside this, molecular taxonomy has been a remarkable tool for the identification of mycorrhizal fungi. Next, please. You can see here, as I told you, that when you are identifying on the basis of morphology, you have to see that what kind of the shape its extending hyphae is taking place. So you can see here, this is why it is funneliformis, because there is the funnel-like structure shape here. Next, please. This is the another species of the avascular mycorrhiza, which is known as Glomus intraradices, while this is the Gigaspora. Gigaspora is having a very unique structure at this point. So there is a bulbous structure at this point here. Next, please. You see here, this is again. So this is the unique feature of Gigaspora. Okay. And these are the another spores like Glomus aggregatum, like Glomus sclerum, Glomus albida, Glomus fasciculatum, Glomus microcarpa, Glomus aggregation, like that. Next, please. Okay. So this was about the mycorrhiza. What is mycorrhiza? What kind of the structures are there? How it is in which part of the plant root it is present. But now the question is, how does AM symbiosis establish in a plant root? How this association takes place? So what are the communications between plant root and mycorrhizal fungi? Why plant is able to give space to get colonized by the avascular mycorrhizal fungi? So all this mystery will be solved. It was solved in 2005 actually. Before 2005, it was not known that what is the molecular communication between plant and root. But a Japanese worker, they cracked this story and they established a proper pathway that how it is taking place. So there are basically, it is divided into 3 phases. One is asymbiotic phase and second is presymbiotic phase, while third one is symbiotic phase. So what is happening in asymbiotic phase? Next, please. In the asymbiotic phase, what is happening? Germination of AM fungal spore takes place. Germinated AM fungal spores respond to host plant root exudate. So host plant is releasing root exudates, which is containing active molecules like strigolactons. So there are 2 molecules basically. One is the strigolacton, which was discovered in and reported in the Japonicus lotus plant, and another is lipocyto-oligosaccharides. So these are the 2 compounds which are basically making the foundation of the establishment of this symbiosis. So strigolacton is a key molecule which initiates molecular dialogue between mycorrhiza, avascular mycorrhiza, and plant. Next, please. So in presymbiotic phase, second phase is the presymbiotic phase. What is happening in this phase? In this phase, the induction of hyphal branching is taking place, which is very, very important. So what is happening? Strigolactons induce hyphal branching and mitochondrial activity in AM plant. There is the release of MIC factors. After perceiving plant signal, AM fungi release bioactive molecules like lipocyto-oligosaccharides, the fungal signaling molecules which induce expression of plant gene like ENOD, nodulin 11, and SYM, RK, CACA-MK, etc. In this way, you can see in this photograph also that there is the metabolic burst, so that hyphal branching is taking place and the activation of certain gene also. Next, please. Okay. So what the E-node 11 is doing, which is known as early nodulin 11, or in short, you can say E-node 11. It is a cell wall protein involved in the cell wall remodeling at the infection site. It is marker gene, not signaling gene. So we should remember it is a marker gene. While the another gene is symbiosis receptor-like kinase, it is a leucine-rich repeat receptor-like kinase located on cell membrane. It perceives lipocytolytic signals. Once activated, it triggers the downstream signaling cascade, including calcium spiking, mutation in SYMRK, block both rhizobial and mycorrhizal symbiosis, showing it controls the common symbiosis pathway. And the another is calcium and calmodulin dependent kinase. It is activated by calcium spiking. Next, please. Third phase is symbiotic phase. So in this phase, what is happening? The formation of proper structures take place. For first formation of episodium or hypanthodium, and the organization of pre-penetration apparatus takes place during this phase, which is a key factor in mycorrhizal infection. Cell wall degrading degrading hydrolytic enzymes are also produced there. Membrane depolarization and calcium spiking taking place. Finally, there is the formation of dichotomously branched tree-shaped structure, which is this, which are known as herbicicules, separated from cytoplasm by periherbascular membrane. Next, please. So this is the electron micrograph of this fungus. I can show you the structure which can you easily see by the light microscope. Next, please. Next. Next, please. Next, please.
Sir, you have to move.
Is it not going ahead?
1 minute, 1 minute, sir.
And
next slide, please.
Sir, 1 minute, sir.
Okay.
We are countering some. Sure.
I'm wondering that why there is no control at my horn. Otherwise,
what happened now? What time it happened?
Coming? Oh. Yeah, next.
it is same slide. Next,
Yeah.
please.
Yeah.
Yeah. Thank you. Thank you very much. So this is the original photographs, which you can see even through the light microscope. These are the vesicles, which are in the cortical cells of the plant root. And these are the vesicles. Next, please. Okay, now let me go. So this was about the introduction and how it is established, how mycorrhiza is making symbiotic association with the plant root. Now we come to the another part of this lecture is that how it is helping under environmental stresses. So as you know that different kind of the environmental stresses, whether it is salt stress, drought stress, heat stress, cold stress, flood stress, heavy metal, or CO2 stress. So it's not possible to cover all these stress in a single lecture. That's why I'm just talking about the either salt stress or drought stress. Next, please. You see, there are the causal factors and impact of drought and salinity on plant growth. So there are the either it is natural cause or human-induced cause. There are several causes which are impacting the plant growth drastically. You can see here that there is the morphological impact, biochemical impact, physiological impact, ultrastructural impact also. In case of the morphology, such kind of the symptoms you can observe, while in biochemical, compatible solutes, antioxidant, osmotic regulation, and all these things are on the they alter during the stress conditions. That's why plant is not able to perform at its high level. Next, please. If I tell you about that, what is the condition of the saline areas in our country as well as throughout the world. So salinity is a condition characterized by the high concentration of soluble salts. It is a complex and harmful threat faced by plants due to disruption of ionic, osmotic, and cell wall homeostasis. The salinization of soil is increasing day by day. At present, out of 1.5 billion hectares cultivated land around the world, about 77 million hectares is affected by excessive salt content. If I talk about the India, about 20% of the total geographical area has been turned into saline and alkaline soil. So these are the black spots are showing you alkaline saline areas. Next, please.
Very heavy soil.
Yeah,
This
I
is
understand.
this is the scenario of the total salt affected area in our different states. So you see that Gujarat is highly affected, UP is about 20%, and Maharashtra is also having a great stake, and other states are having salt affected areas, but not like UP, Gujarat, and Maharashtra. Next, please. This is the condition of the world. So you can see here that Asia, Pacific, and Australia, Europe, Latin America. This is North. I think there is some mistake here. So North America and Africa, all these areas are having salt affected. Next, please. How and when we can say that there is this soil is saline. So if soils are therefore soils are classified as saline when their electrical conductivity is 4 decisiemens per meter or higher, which is equal to approximately 40 millimolar NaCl and generates an osmotic pressure of about 0.2. Next, please. This is another slide which is showing you category of saline solutions, their application and impact on the plant growth. So you can say that the soil is non-saline when the electrical conductivity is like this, and slightly saline at this, moderately saline, highly saline, and very strongly saline. Next, please. Genesis कैसे बनता है? How the soil salinity is taking place? Soil salinization may be the result of natural or man-made factors. Like, what are the natural factors? Natural factors may be like weathering of the rocks, deposition of arsenic salt, topographical factors, groundwater, table fluctuation, salt lakes, etc. While the man-made, like irrigational water, there are certain areas where the water is in fact having very high salt, that is also known as khara pani, and use of chemical fertilizers and pesticides. Khara pani is not a very broad, widely accepted term. It's a local term. Generally, in the states like UP and Haryana, they are calling it khara pani. Next, please. So this is what is happening sometime. Actually, we generally use a term physiological drought. Salt stress causes physiological drought. Why the physiological drought? Because water is sufficient, but plant is not able to uptake that water. That condition is known as physiological drought condition. So why it is happening? Because due to all these procedures, the salt is accumulated at the in the vicinity of the root. That's why there is the change in the osmotic potential. Therefore, plant is not able to take up this water. Next, please. There are the human-induced soil salinization in the different parts of the world. And you see here, Asia is highly affected. Next. How does salinity affect plant growth? There are several reasons. One is the physiological drought. I just told you, excessive toxicity of ions to the cell, high uptake of sodium and chloride ions. There is the reduced because of this, as I told you, there is the change in the osmotic potential. Because of this, there is the disruption of cell organelles and their metabolism. And because of this, there is the nutrient imbalance in the plant. That's why ultimately there is the reduced plant growth. Next, please. What are the potential solutions? As I told you in the beginning of my lecture, that there are several procedures, but these procedures are not feasible at the economic scale also. Identification of new salt-tolerant crop can be an option. Development of salt-tolerant crops through breeding and genetic programs can be an option. Leaching of excessive salts or desalination seawater for use in irrigation can be an option, but very costly. And another option is application of soil microorganisms such as mycorrhizal fungi, Piriformospora indica, or plant growth promoting bacteria can be a good option. Next, please. What kind of the protecting mechanism these soil microbes are playing? So you can see whether it is plant growth promoting bacteria, AM fungi, P. indica. So they are helping the plant in different way, whether it is the increased gaseous exchange, increased photosynthetic rate, high antioxidant production, nutrient balance. All these things are these microbes are helping in all these ways. Next, please. So the question is why we should use it? Why arbuscular mycorrhiza? Because arbuscular mycorrhiza has been the role of arbuscular mycorrhiza has been well established in playing certain roles, like it alleviates osmotic stress, it modulates soil carbon sequestration, influence soil particle aggregation, maintains soil fertility, induce disease resistance, and therefore increase plant growth. Next, please. You can see in this slide that osmolytes accumulation and nutrient acquisition in mycorrhizal plants under abiotic stress has been reported as well. So whether it is proline, whether it is trehalose, glycine betaine, mannitol, sorbitol, inositol, like all these osmolytes. So mycorrhizal fungi has been reported to play significant role in altering all these kind of the osmolytes under stress condition, particularly salt stress and drought stress. Next, please. Not only this, their role has been established in the production of enzymatic antioxidants and non-enzymatic oxidants like all these. Next, please. I'm taking a little faster because I see that there is the limited time now. It's now 3:30. So aim confer increased plant growth and biomass, because increased biomass may be due to greater nutrient exploration capacity of extra radical hyphae of the mycorrhizal fungi, even under stress conditions, whether it is drought stress or salt stress. Next, please. Nutrient acquisition, because it is also having its control over certain kind of the transporters. It plays certain roles there. That's why it has been established, or it has been revealed, that this fungus is playing very critical role in nutrient acquisition. Next, please. Improved ionic balance, sodium and potassium. So you see that there should be a proper balance in that. If there are high concentration of sodium, it is harmful for the plant cell. So mycorrhizal fungi has been found to make a proper balance between sodium and potassium. So higher sodium potassium ratio disrupt ionic balance in cytoplasm, but if it is reverse, then it will be beneficial for the plant. So mycorrhizal fungi has been found to make it like increased potassium sodium ratio. Next, please. It modulates osmolyte and antioxidant. I have covered this part in the previous slide. So whether it is SOD or whether it is total sugar, its role has been found in all these. Next, please. Effect of AM fungi on alpha tocopherol content. Alpha tocopherol content also has been found to be altered because of the mycorrhizal fungi, and it is very, very important. As you know that alpha tocopherol acts as the main lipid soluble antioxidant in chloroplast and protects photosynthetic membrane by scavenging, singlet oxygen, and stopping liquid peroxidation chain reactions. Therefore, prevent oxidative damage and preserving photosynthetic efficiency and water content in the plant. So this is very, very important. Next, please. Enzymatic activities like catalase, we have found that in mycorrhizal plant, the activity of catalase is also altered. Next, please. Ascorbate peroxidase activities. So all these enzymes have been studied not by us only, there are a lot of research papers which are showing that under controlled conditions, as compared to the controlled condition under inoculated conditions, the condition of the plant has been far better, far better. Next, please. Melandihalide MDA activity in the plant has also been reported to be altered positively by the mycorrhizal fungi. So you can see in this slide, this MDA content is remain even at the 200 millimolar salt. You will see that there is the what is the happening in case of the mycorrhizal fungi. Next, please. Relative permeability, it is also changed. Next, please. Okay, let me come to this. This is the evidence that what is happening when mycorrhizal fungi is in the plant root. You can see here. So this is basically energy dispersive X-ray analysis. You can see here that at 200 millimolar, that in case of the mycorrhizal plants, there is the accumulation of this salt has been found to be comparatively lesser as has been reported in case of the non-mycorrhizal plant. Next, please. Yeah, this is another slide, same thing. Next, please. Okay, let me explain this about this. This is the electron micrograph of the plant which was growing under salt stress condition. And you can see here that at 200 millimolar salt stress, in case of the non-mycorrhizal plant, you will see there is the widening in the space of the cell wall. While this space is not seen in the case of mycorrhizal plant. So this shows that membrane integrity is remain intact when mycorrhiza is present in the plant root. It is helping there. Next, please. As far as the chloroplast condition is concerned, we also reported better results in case of the chloroplast when the plant were growing in the salt stress conditions. Next, please. Okay, let me explain this again. This is the formation of plastoglobules. You see here, this is 100 millimolar salt stress here. You see what is the condition of plastoglobules are these. These are the plastoglobules here in the control plant. And you see here, but if you see at 200 millimolar, compare this and that. When 200 millimolar salt has been given to the plant, there is the less number of plastoglobules as compared to the mycorrhizal plants at the same concentration of the salt. So plastoglobules are the site of the synthesis of alpha tocopherols. If there is the bigger and large number of the plastoglobules, it gives an indication that the plant photosynthetic framework machinery is working finely under stress condition when such microbes are colonizing the plant root. Next, please. Next, please. So this is my concluding slide almost, that what kind of the actions under salt stress mycorrhizal fungi is taking place. So this is a control plant which is growing under salt stress condition. This is the another plant which is inoculated with the mycorrhizal fungi. So you can, I have summarized it in 5 points. One is the photosynthetic protection, which was far better in case of the mycorrhizal plants. Antioxidant production, which was better in case of the mycorrhizal plants. Osmotic adjustment, also modulated in case of the mycorrhizal plants. Water homeostasis was finally reported better in case of the mycorrhizal plants. And 5th point is ionic balance. So there was the increased potassium sodium and magnesium sodium ratio. Yeah, it's okay. Next, please. So how we can conclude it? We can say that AMF provides physiologically more adaptive mycorrhizal root system, meet initial requirements of water and nutrient, modify rhizosphere chemical properties, maintain ionic balance and manage accumulation of antioxidants and osmoprotectants, minimize shrinkage of protoplasm and widening of apoplastic space between cell wall and cell membrane. Therefore, we can state here that use of AM fungi or mycorrhizal fungi may be a potential approach to improve plant growth and productivity under salt stress conditions. Next slide, please. Thank you very much for your patient hearing and inviting me for delivering a lecture in this prestigious FDP program. Thank you very much, all my colleagues.
Thank you, sir.
Thank
One
you.
question that I think we have in chat box. Let me
Ji.
find out. So try to see that. Dr. Payal,
Yes, sir.
you may lead and see the chat box, please.
Thank you very much, sir, for your highly insightful lecture. Your valuable perspectives have greatly enriched our understanding of these important research topics. We shall now move on to our question and answer sessions. Participants are requested to post their questions in the chat box.
One question I saw that in chat box that relate with the mycorrhiza.
You can once this user accept your no.
No
Doctor,
question.
आप पढ़ दीजिए, मैं दे दूंगा. Chat box is there.
Sir, I'm reading. Sir, the question is asked by Saurav Tripathi, and he asked, recently, spatial transcriptomes are widely used in biotechnology by our neighboring country like China to quantify the variability. Either it is due to biotic or abiotic stress. So, sir, is there any recent studies were made for mycorrhizal context? As we know that spatial transcriptions can able to explore variability, cellular or even subcellular localizations. Can you put your insights into this context?
As far as my knowledge is concerned, I'm not having a very recent report about that. But I will tell you one thing. The mycorrhizal fungi is a very good fungus. It is helping plant in a remarkable way. But the major problem with this fungus is it is growing with the plant only because it is an obligate symbiont. So whenever we are trying to do the studies at the molecular level, we need a monoculture. We need a pure culture. Until unless we are not having a pure culture, such kind of the studies cannot be taken place. So there is the technology which is root organ culture technology. There are certain laboratories which are doing this technology and they are into that kind of the research also. But as far as your question is concerned, I am not having the latest information regarding this. Thank you. Next, please. Next.
Dr. Enrico, do you have any suggestion, query, sir?
Yes, thanks. I have a question.
Yeah.
Have you any data on differences between wheat field and or annual crop and
Sir, please, can you speak a little louder, please?
my voice is going up and down. Sorry.
You can
I was
keep your
asking
mic little closer.
for differences between the effects of mycorrhiza on a crop like wheat or a crop on a fruit garden. Annual
crop.
crops and permanent crops.
So
different effects on annual crops and perennial or multi-year crop.
Okay, if I'm getting your question rightly, actually mycorrhiza is not a host-specific fungus. So the beauty of this fungus is it is not host-specific. It is also performing in the annual crops. It is also performing in the biannual crops or herbaceous plants. So there is no host specificity, whether it is a crop like wheat or a fruit crop, which is a perennial crop. So reports are saying that it is like citrus. There are the Chinese groups which are working on the citrus trees. There are wonderful results. And in fact, in India also, people have worked on annual crops like wheat, maize, and rice. So its performance has been reported in all cases, sir. Whatever.
My point is on soil disturbance, effect of soil disturbance on the survival of a mycorrhiza and the effect on the following crop.
Oh, okay. So you mean if there is the disturbance in the soil, there is the any kind of
Right.
impact on the mycorrhizal fungi? Is it?
Right.
Yeah. Oh, yes, of course. Because what happening? If there is the disturbance in the soil, plowing or something like that, the network, common mycorrhizal network is disturbed. Because of the disturbance of the common mycorrhizal network, of course, the performance of the mycorrhiza is also affected.
Thank you.
Thank you.
Okay. Okay, Payal, thank you, sir, for nice, insightful talk, right? And so grateful to you.
Thank you. Thank you very much, DK. Thank you. And everybody in the panel, thank you very much. And my all colleagues, all participants, thank you. Thank you very much.
Right. Payal, may please
Yes.
lead.
Thank you, sir, for your enlightening presentation and for patiently responding to all the queries. Your valuable insights and expertise have made this session both informative and inspiring. On behalf of the organizing committee and all the participants, I extend our sincere gratitude for your time and for sharing your knowledge with us. Thank you once again, sir.
Thank you. Thank you. So nice of you. Thank you.
With this, we come to the successful conclusion of session 2 and the second day of our faculty development program. Today's sessions provided valuable insights into climate resilient agriculture, weed improvement, plant microbe interactions, soil health, and sustainable agricultural systems. We were privileged to learn from our two eminent scientists whose contributions have significantly advanced agricultural research and innovation. I'm sure that the knowledge shared today will inspire new ideas, collaborations, and research directions among all the participants. On behalf of the organizing committee, I once again extended our heartfelt gratitude to our eminent speakers, Dr. Arun Kumar Joshi and Dr. Bhupender Giri, for their enlightening lectures and for generously sharing their expertise and experiences with us. I also thanks all the participants for their enthusiastic participation, thoughtful questions, and active engagement throughout the day. As we conclude today's program, I would like to remind all participants that we will reconvene tomorrow for day 3 of the Faculty Development Program, where we will continue our academic journey with another exciting theme and a lineup of eminent experts. We encourage everyone to join us on time and actively participate in the sessions to make the most of this learning opportunity. Thank you all for all your presence and participation. We look forward to welcoming you again tomorrow. Have a pleasant evening and thank you all once again.
Thank you. Bye-bye. See you tomorrow.